Good morning. Okay, welcome to day 2 of the BE Innovations Workshop. It's great to see people in the room. I see people are still making their way to their tables. Good morning, good evening, good afternoon for those of you online. Just as a reminder, we will send each of you all the registrants an email within the next 3 weeks with regard to all the notes being online as well as all the videos being online. Maybe just speaking on behalf of myself, I've really enjoyed the meeting. I've been to many BE meetings over the years, and yeah, it's really quite inspiring. I think the new ideas that at least I heard in terms of bioequivalence. And we're very fortunate to have some of the moderators from yesterday present on the 3 topics that were discussed in the in-person session yesterday afternoon. And Jihong will first give us a summary of the first topic on approaches for oncology products. Jihong.
Good morning, everyone. Yesterday, for oncology product session, we have 3 tables. Each table have a specific topic category title. First table covered the study population. Table 2 covered patient B study design. But yesterday we decided to consolidate table 1, table 2 questions because they are very related. And question 3, biowaiver and alternative approaches. So let me first summarize what we discussed from the first 2 tables, study population, patient B study. On the study population category, we pose question whether they have any input or any suggestion from their experience for that flow chart for study population selection. And for PKB study in patient for oncology product, we also pose a question, what are alternative approaches feasible to replace conventional steady state studies? And we also ask any suggestion for addressing high-risk product in patient PK study. So we have some interest folks and one person from academia. So initially, the industry folks wanted to clarify some, procedure from the FDA process, PSC updated process. So yeah, that is one of my talks. So I provided some detail of the process that agency practice with some detail. Yesterday presentation, yeah, I could not disclose some, but on the table I could give a more little detail that we usually practice. So industrial attendees discussed some challenges when they conduct the B study in patients. So many items, patient recruitment issues, for example, patient population for rare disease or narrow therapeutic indication product. So patient retention issues. So especially when studies are conducted, a patient in more advanced stage cancer, given that the short life expectancy, there might be a lot of challenge to retain a patient during the study, before, after. So that is another difficulty. And those modification issues during the study and concomitant medication issue. Even crossover design, even parallel design, concomitant medication issues, yeah, always, serve as a compounding factor. So FDA attendees acknowledge all difficulties. So probably, we would, come up with some the plan to look at our PSC, try to give more clarity, which could help the industry. Industry attendee also inquire regarding enrollment of patient with non-approved cancer type of product with a narrow labeled indication. But from FDA side, we still have some more no room to allow that. But sponsor can propose some alternative approaches with some exploration. We definitely consider that type of approach. So some key takeaway from our team summary. FDA side, we continue to evaluate industry proposal for any alternative B study design by ensuring transparency and consistency in decision making for study population changes or update. Agency also consider providing additional clarity or guidance on the procedure pathway for reassessing patient-based PSG to help industry navigate the process more efficiently, including concomitant medication, more feasible option for study design. So industry group are welcome to submit the proposal of any alternative study design through the appropriate regulatory channel with a relevant scientific justification when seeking the assessment of patient-based PSG. They will also advise to limit patient enrollment to approve cancer indication in B study in alignment with the FDA guidance. They were also encouraged to develop the mitigation strategy to address patient recruitment and retention challenge in the B study, particularly for rare disease or when they encounter some challenge to recruit a patient population. So this is some summary from table 1, table 2. Let me move to table 3 question group. Under table 3, which cover biowaiver alternative approach for oncology product, for this group, they discuss 3 questions. First question was viable, non-viable approaches to waiving in vivo testing for oncology oral product. And second question was research, what research gap are to address this biowaiver for oncology oral product. And third question was a key factor to ask a key factor for successful PK/PD modeling as a supportive evidence for biowaiver. They provided a 3-page script, so I couldn't read all script, but some key takeaway from that discussion group. In order to substantiate a biowaiver justification, experiment design and product characterization activity should be integrated into generic drug development program from the beginning, rather than incorporate after the fact. Case by engagement with agency through pre-submission meeting and control qualification meeting is recommended for novel or out of guidance approach, such as dose normalized PK bridging across strengths or waivers for BCS2B, BCS4 oncology product. Advancing the predictive accuracy PBPK model and which alternative approach are appropriate will require coordinated research effort among the industry, academia, regulatory agency, supported by larger shaped data set. This group make a very interesting note. The term biowaiver have a distinct regulatory implication and warrant careful the application. In the many oncology contexts, terminologies such as alternative approaches or additional supportive evidence may be suitable. So this is the summary from the 3 table for oncology product session. So, personally, I was so grateful to have all industry folks for this discussion. We could, hear the voice from the field I think at least, clinical team and DQMM, QCP team may come up with some idea to look at our PSG or our submitted data. We may come up with some plan or clarity to improve, our PSG, which can help the industry to develop the oncology product. Yeah, that is summary.
Okay, thanks, Dr. Sung, for that summary of oncology that was discussed yesterday afternoon at the breakout sessions. I was at table 3 talking about bio waivers of oncology, and I'll just add that we included some of the what came up in some of the conversations were some of the bio waiver frameworks that were discussed with the nitrosamines panel also, which is topic 2 yesterday, and Dr. Cheng will be talking about nitrosamine-driven reformulation and bioequivalence discussions from yesterday afternoon.
Thank you, Dr. Polli, for the nice introduction. Good morning, good afternoon, and good evening, everyone. So I will provide a brief overview of what we have discussed for the 2 tables. We actually integrate 2 tables into 1 table for the yesterday discussion, but we really enjoyed the discussion yesterday. And please bear with me. I will probably just read through the slides. So our topic 2 is regarding the nitrosamine-driven reformulation and the bioequivalence. We have discussed over the 4 questions. So you already have the questions we discussed yesterday, so I will just not repeat the questions. So the first question is for the IR products, nitrosamine-impacted IR products. And here we try to propose the risk assessment framework. The questions we discussed, we also put a threshold that we would like to go over and welcome the opinions, whether this threshold is feasible and how we can adjust the range. However, we don't have a conclusion yet, but the current thinking from our table is that this threshold is feasible, but we may need more using the critical parameters, dose number, and the bioavailability. These thresholds are feasible, but we need more data to adjust the threshold before we can roll out these thresholds for more broader applications. And for the best practice, how we came up with the risk assessment framework, we came up like step-by-step approach. The first step is that we will submit the multi-pH dissolution study, pH 1.2, 4.5, and 6.8, plus the dissolution conducted on the QC media. And we also need to confirm the AC phase stability up to 2 hours or more than 2 hours. And the third approach is to use the PBPK, PBBM modeling if the solution is inconclusive. And the last approach, if the prior approach are not like feasible, the company will propose doing the pilot PK study and come to the full bioequivalence study. And for the questions we discussed for the modified release products, whether we can extend the IR guidance to the NR products. The short answer is yes, but we need to evaluate like in a case-by-case basis. What additional condition? Under what conditions are these extending IR guidance feasible for the NR products? We need to first make sure that the the changing limited to the antioxidant and pH modifier within a certain amount, say 10 milligrams. And we want to make sure that the release profiles is demonstrated unchanged compare pre and post change formulations. We will also want to make sure that the release mechanism is not altered. And finally, it's an interesting discussion that people think the DR products are actually closest to the IR products. So that means the IR guidance currently we rolled out probably can be applicable to the DR products, but not other extended release products. And what we put like additional thought about the additional consideration for the modified release products, we would propose the multi-stage dissolutions. And we also want to make sure that the dissolution testings are conducted in the biorelevant media. There is some discussion like the alcohol dose dumping evaluation is needed where relevant. We also want to consider the release mechanisms such as matrix, osmotic, reservoir. And for the modified release products, the PBPK, PBPK modeling are highly recommended. And for some sponsor, depending on the resource they have, sometimes the pilot B study may be faster or easier or more straightforward for the company. And then I move on to the question series. Here we are discussing the alternative B approaches. For the question series, the group proposed the stepwise evidence hierarchy. The tier 1 will be the fundamental like laboratory works. We proposed like to conduct the multi-pH dissolutions plus the QC media dissolution testing. And here the group also proposed like we need to make sure like the low nitrite excipient selections and to assess the packages. Here there's a common like discussion about the predictive stabilities and the stability also under the acid phase up to 2 hours. And for the tier 2, the group proposed the predictive modeling. They will integrate PBPK, PBPM modeling plus by incorporating the dissolution profiles. And people are agreed with QC dissolution alone often is insufficient. The dissolution testing conducted on the biorelevant media and the mechanistic understanding about the drug release is necessary. The tier 3 approach is that to integrate the in vitro and the mechanistic tools such as CACO2 cell permeation assays, the transporter interaction studies, the animal studies or organ-on-chip systems. And the group also think it's useful for BCS3 or for nitrosamine peptide products, along with the permeability sensitive products. And with the tier 3, we can bridge the gaps between the dissolution and the 4B study. The tier 4 will be like similarly conduct the pilot PK studies and the full clinical B study. What are the additional considerations for these alternative B approaches? We need to develop the more flexible products. We need to compare the post-change versus pre-change formulations under the BE study. There's also some challenge that for the earlier approved products, we may not have the IOD on the market available. And here is interesting that The group also discussed the DR versus the extended release products. So the conclusion is similar, like simple enteric-coated DR products act more like the IR products. And the group also think about like the shoe pack need to be updated. So for the question 4, the key challenges and the proposed solutions. We discussed over like the CMC and the bioequivalence review need to be integrated among the multiple disciplinary. And people also discuss about like the harmonization about the different agency is ongoing, which also pose a challenge to the industry that they want to have a guidance, like consistent guidance to follow. And another discussion around these challenges and the future needed, we want to develop a guidance like beyond the BCS classification. People are thinking that BCS classification are too strict, and we want to add the formulation complexity layer beyond the BCS classifications. And the research priorities, as I mentioned before, we need to validate the dose number and the bioavailability threshold across different products. And people are thinking that shared excipient nitrite database is helpful to help industry to conduct the reformulations. The key takeaways of these 4 questions would be, the first one, we would like to come up with the stepwise risk-based evidence hierarchy. The first step is to, as I mentioned, fundamental testing, solubility, permeability, and the dissolution testings. And we can use the mechanistic modeling, PBPK, to combine with the intermediate tools. The final approach would be conduct the pilot PK study or the full bioequivalence study. Second, BCS4 drug substance subclassifications like BCS2 like solubility, permeability, solubility limited, and BCS3 like solubility limited. We can use the threshold, dose number, and the bioavailability, but we need to verify this with more researches. And the IR guidance we can be extended to the MR and the DR products on a case-by-case basis. And we also think the DR product is more close to the IR products than the extended release products. And also the group thinking the DR should not be grouped with complex NR products. We need to integrate the CNC and the bioequivalence review across different disciplines to come up with the common guidance. And we also would like to harmonize among different agencies. The formulation complexity should be layered on top of BCS classification. SUPAP modernization is needed, and share the database for the excipients and published case studies are essential to move these projects forward. Thank you.
Okay, and last but not least, topic 3, Dr. Boyce will summarize discontinued RLDs. But before I forget, I just want to thank all the folks in the room. I want to thank yesterday's table moderators and scribes. Also thank Sam Rainey, kind of helped organize these sessions. To me, they're like, as much as I enjoy the presentations, actually, the novel discussions from the presentations, really enjoyed the table discussions too. Okay, Heather?
Okay, good morning, everyone. So I am up here summarizing the discussion for topic 3, which was on the discontinued RLDs with no reference standards available. I want to give a special thank you to Dr. Shamima Nasrin, who compiled some wonderful notes that I will be summarizing for everyone today, and also Dr. Gerald Sal and Dr. Sivacharan Kollipara, who have and Dr. Robert Hopeful, who moderated the sessions yesterday. And so I think yesterday we gained a lot of really important insight on this topic. I think a lot of times working at the agency, we receive a lot of complex correspondence and inquiries regarding, how do we get our products approved using alternative approaches. And one of the big questions we might have in the agency is why not just pursue a 505(b)(2) pathway? It would seem like an easier solution for companies trying to bring back products. And I think that what we heard yesterday was the industry consensus was very clear that generic companies just aren't built for the commercial and regulatory demands of the 505(b)(2) pathway. And the 505(j) pathway does offer a lower cost for applicants and is a lot simpler to execute on their commercial product development strategy. And also, most importantly, the 505(j) pathway offers automatic pharmacy substitution, which is key for market entry. And so the industry was very clear that the 505(j) pathway is preferred, even though there are substantial hurdles with this particular type of alternative bioequivalence pathway. And I think the FDA also confirmed that even though the hurdle to approve products without a reference standard can be high, especially for that first applicant trying to get that product approved, that FDA confirmed we do have these bioequivalence approaches and we are willing to work with industry on them, and that we think these BE approaches can legitimately exist within the current 505(j) framework. And I think that that was one of the topics that was a very contentious debate was, and what kind of scenarios are these legitimate 505(j) pathways acceptable? And I think the groups broke down the 2 types of comparators. We have simple cases where you have the same dosage form, where transitivity is obvious, and that those situations make the BE assessment more straightforward. There are, however, other scenarios that are much more complex, where the dosage form is not the same dosage form. So IR versus XR, as we saw in the potassium chloride example, for instance. So these can be especially challenging. And, you just might not have like the historical data to complete the totality of evidence approach. And the target profile, comparative profile might not be available for this. And this can also increase cost and time and uncertainty when an applicant might be trying to develop a product utilizing that pathway. And while there was acknowledgement that PBPK modeling can provide direction, it's not always a substitute. And especially in vivo pilot data is still potentially required to overcome those hurdles. And then a reoccurring theme that came up during the discussion is that while public data is available, especially at drugs at FDA in the summary data, the overall depth might not be sufficient to produce reliable models. And I think this is where participants expressed interest in this idea of master data files. So sort of like what Dr. Brooke inferred yesterday with her presentation, how do we create model data banks of information for these reference products that don't physically exist on the market, but we have all this data to support that. And then some of the key requests that industry asked to overcome some of these challenges was, there was a lot of discussion on non-US reference products. So there were discussions that arose around using European Union source products as alternative comparators, and precedent from biosimilars. Discussions came up about that, how biosimilars have leveraged that using analytical bridging between the EU and the US product. And then analytical compares, again, how do we extend that to smaller molecules? And there were some real cases that were brought up that were specific for a German specific product. And then I think overall, the request from industry was that FDA does provide more clear frameworks for selecting the alternative comparators, and that we strongly encourage applicants to engage with the FDA early through our various meeting request pathways. So the pilot model informed approach, the product development meeting requests and control correspondences are all wonderful avenues that you can engage with the FDA, and applicants have noted that those do help improve the efficiency of their product development program. And thank you so much for everybody who participated in these discussions yesterday, and I hope I covered everything. Okay, thank you.
All right. Good morning, everyone. And good afternoon, good morning, good evening to everyone joining us online. Yesterday was a really heavy lifting day, huh? I think you guys got the most important kind of drop down yesterday. So hopefully we have a bit more like a relaxing day today. But I wish you had really enjoyed the discussions and the presentations the first day of the workshop. So yesterday we covered several critical and complex areas that shaping the future of generic drug development. We began with the bioequivalence approaches for oncology oral products, where discussion highlighted the need of balanced ethical considerations with scientific rigor, often requiring alternative approaches and clearly defined decision frameworks. We then explored the nitrosamine-driven reformulation, where we emphasize risk-based assessment, and the modeling approach, the totality of evidence to support bioequivalence. We also addressed the challenge of discontinued reference list products and available reference standards, and the innovation strategies being discussed overcome these barriers to generic development. So all of those sessions, a common theme emerged: the increasing importance of mechanistic understanding and integrated evidence in supporting regulatory and the development decisions. So this brings us to today's session 5, titled, "When is dissolution truly biopredictive?" I think this is a question sit at the heart of lots of the challenges we discussed yesterday. So dissolution testing has been a cornerstone, or is a cornerstone for the quality control. However, increasingly, we are asking more of it. we want to explore the potential to serve as a predictive indicator of in vivo performance. So this raises important questions, right? Under what conditions can dissolution data reliably inform by equivalence? And when can it meaningfully contribute to, or even in certain cases, replace in vivo studies? So in this session, we will examine biorelevant and biopredictive dissolution approaches, the discriminating ability of dissolution methods and how that is related to tell different clinical performance, and then how these methods can be strengthened through integration with the modeling and the simulation framework, such as PBPK and PBBM. Of course, we have to consider the critical issue of evidence sufficiency, what constitutes adequate support for dissolution-based bioequivalence, and discuss both the opportunities and the limitations. Ultimately, this session aims to advance a shared understanding of how dissolution can evolve from a quality tool into a fit for purpose by predictive component within the broader bioequivalence framework. So with that, I'm pleased to welcome you to the session 5. I'm really looking forward all the discussions with our audience. This session includes speakers, including myself, and then followed by Dr. James Polli, who will share stories from 2 projects on dissolution methods for products with varying biopharmaceutical risks. Dr. Sivacharan, Kollipara will address the fundamental question, what is a true biopredictive dissolution method from an industry perspective. Dr. Deanna Mudie will discuss the best practice and the challenges in establishing fit-for-purpose dissolution test, particularly when integrating with PBPK modeling. Finally, Dr. Ahmed Zidan will provide insights from a case study on number changing to illustrate predictive dissolution and also modeling approaches. Following the presentations, there will be a Q&A panel, so please have your questions ready. We're hoping to have a very engaging discussion. So with that, I'm going to maybe start with the 2 housekeeping logistics. I'm not going to introduce our speakers' bio. You can find their detailed bio through the CRCG website, the workshop website.
Okay, all right. What I'd like to speak with you about is really just 2 topics. One dealing with IVIVC, and what I'd like to do is mostly focus on, you know, from a dissolution standpoint, what was going through our minds to achieve this IVIVC for a high-risk product. And I attended the meeting last week where there was some discussion about a new framework, and one of the topics was high biopharmaceutical risk. And I think I understood that, you know, it'd be good to have a PK available that was consistent with formulation understanding. That's one topic. Another topic is deconvolution-based analysis for lower biopharmaceutical risk products. Okay, so those 2 things I'll talk about. With regard to the second topic, you know, just one question I always have in the back of my mind is, do we have routine best practices for data analysis as it relates to the role of dissolution in product understanding? And I kind of think the answer is no, actually, which is why I'm showing some of these analyses, which I think are actually quite simple. Okay, so the first little story about itraconazole, we fabricated itraconazole amorphous solid dispersion tablets for the purposes of doing IVIVC. We're just kind of interested in some formulation understanding. These are relatively complex formulations. Some of the findings were we were able to do a level A IVIVC successfully with fast, medium, and slow. We also had an oral solution as an arm. The largest contributor to the overall effect were polymer grade, which I understand may not be a confusing issue in the context of manufacturing, but we were interested in polymer grade issues. The polymer was HPMC AS, and we had L grade, which provided better results, so to speak, than M grade. The tablets were dry granulated, so we had issues of slugging. We had variables of slugging and tablet compaction pressures, as well as disintegrator amount for those tablets that use HPMC AS M grade. Those were also factors, not the largest factor though. And the thing I'd like to emphasize is that for the medium and slow formulations, their PKs were more similar than what we would have anticipated from preliminary dissolution alone. And then that, of course, inspired post-clinical study, revisiting dissolution and trying to push the slow towards the medium. And I'll show you how we try to bend those curves in the laboratory. And what we ended up doing was actually grinding the tablets as a part of the dissolution method. The slow formulation was sort of just too slow, particularly in the USP apparatus 2, where there's not a lot of agitation. In vivo, it's well known, in fact, quite measured in terms of shear forces. So we thought that was a reasonable thing to do, particularly given the PK data. And well, I'll certainly share it so you can have your own opinion. So here's a preclinical dissolution data. HPMCAS is an enteric polymer, so we needed to use this. It doesn't dissolve at all at the lower pHs. And I guess the thing I'd point out is the slow formulation was, reasonably slow compared to the other formulations, which in vivo did not manifest as much, although it was slow. And just to remind you, again, the scope was try to do an IV-IVC, level A IV-IVC per the guidance, which says IV-IVC is a mathematical model correlating the entire PK, in vitro profile, like dissolution, with the entire PK profile. And then here's sort of the thing that, at least from a modeling standpoint, one is interested in doing in terms of predictions of the PK profiles in terms of AUCs with predictive accuracy, so to speak, for fast, medium, and slow formulation. So that worked out. And in terms of how we got there, I guess the thing to focus on here is we actually did quite a bit of in vitro dissolution post clinical study. ASDs, I know many of you work on ASDs. It would be great if we knew what dissolution we could always use that gave us the right answer. Unfortunately, there's not a universal test for ASDs, at least. And so we had to do quite a bit of in vitro dissolution after the study to get this to work, so to speak. And we think that did provide some additional insight into ASD dissolution performance. But yeah, there's still a lot there, I would have to say. So this is a schematic of tablet manufacturing. It was a dry granulation method. So we did spray dry dispersions of drug and polymer, did dry granulation and tableting, making fast, medium, and slow formulations. And here's some details of the tablet characteristics in terms of fast, medium, and slow. 20% drug loading. We use sodium starch glycolate as disintegrant, and again, polymer grade L for fast and M for medium and slow. And again, varying slugging pressure and tablet compaction pressure. And here are resulting PK profiles, along with an oral solution in a four-way crossover study. So this is sort of almost like a generic drug. I mean, this drug is certainly available generically. In the literature, there's highlighted in red there is a really nice study where they studied the IV as well as the oral solution. And our oral solution actually was very, very similar pharmacokinetically to the literature. So we're able to have some good basis for the IV-IVC in terms of how the drug, its disposition post-dissolution, including through the use of the oral solution in this particular study with these subjects. So itraconazole and ASD is presumably absorption rate limited by dissolution. So we did deconvolution analysis. The drug is known to be two-compartment. And here are the Lu-Rigomin deconvolution profiles of the oral solution as well as fast, medium, and slow. These then became our target dissolutions. So again, the Lou Riegelman deconvolution profiles are on the right-hand side. On the left-hand side, we paid a lot of attention to the extent of dissolution. So in this approach, we're assuming that dissolution is incomplete in vivo, resulting in differences in AUC in vivo. And we wanted to mimic that. So we didn't, again, with dissolution being rate limiting, presumably, we were willing to have incomplete dissolution, and we knew the extent of that incomplete dissolution. And that, so just relating the relative bioavailability compared to the oral solution, compared to the AUCs for fast, medium, and slow. And then HPMCAS is an enteric polymer, so the student doing all this work, she changed pH at every 0.2 pH units and did a lot of other things in terms of agitation rates and grinding tablets or not grinding tablets, arguably trying to mimic the in vivo conditions that gave the in vitro results, in vivo results. Okay, here's a picture of her in the upper right-hand corner, like finally succeeding and celebrating that she was able to get the right curve. And on the left-hand side are the resulting dissolution profiles, which involved using apparatus 2 at 50 RPMs, a certain pH, and tablets again were ground. And what that did is that they see the green profile there. It made the slow formulation much faster. It actually had no effect on the medium formula, on the fast formulation, or not even much effect on the medium formulation. So we're able to achieve the target profiles that we were looking for by presumably doing something more biosimilar, more similar to in vivo. And then on the right-hand side, those same dissolution profiles, the colored profiles are plotted there. The x-axis is just every time point that we took. It's not linear. And then showing the deconvolution profiles that you saw earlier. So you can see there is a time difference there. Overall, dissolution was faster than absorption. And the IVIVC guidance allows for a time correction factor. So we just couldn't get it all the way we wanted, but we were generally happy though. So this is on the right-hand side, just is showing the, it's really fitting, not so much predicting, fitting the oral data to a 2-compartment drug. The drug's known to have its 2-compartment pharmacokinetics. So we have a nice basis for the disposition of the drug post-dissolution. And then all we did was feed in the fitted dissolution profiles, which we knew would work at this point. So then just doing that, we did have to come up with a correction factor for time. So we did slow down dissolution in vitro for an in vivo component. And then that resulted. So these are actually real predictions. Again, very much a big reliance on the solution, as well as just knowing the drug was 2-compartment drug. Again, I would just go back to the dissolution profile that the deconvolutions gave us a target. And the thing that was sort of key was grinding the tablets. So I don't think these tablets are, I don't think their release mechanism is based on erosion, unlike maybe some other types of ASD formulations. I think it's a little more complex than that, unfortunately, which we're still kind of studying. But in any event, we do think this did work for this formulation family. So that's a bit about IVIVC of a high-risk product in ASD. What I'd like to next talk about is sort of lower risk, where using, again, emphasizing deconvolution. Again, I think you can learn a lot by plotting deconvoluted absorption profiles and dissolution. So here is fraction absorbed and fraction dissolved of Norvir, ritonavir ASD. And the upper curve is the dissolution profile, which employs a very high concentration of surfactant. And then below that is the deconvolution absorption profile. And it starts making you wonder, like, is there too much surfactant in this? Is it dissolving too quickly? This sort of thing. But in terms of maybe something I think is actually helpful for some sort of biopharmaceutic insight is is sort of the classic Levy plot, fraction absorbed versus fraction dissolved. And here's something we derived quite some time ago. And the model behind this is very simple. In fact, it's overly simplified, right? It's a first order dissolution model with first order permeation. And then, of course, absorption and dissolution are time dependent. And if you isolate those, what you get is at the bottom there, you get fraction absorbed as a function of fraction dissolved, and that increases. And the nature of that relationship depends upon this alpha term, which is simply the ratio of the permeation step to the dissolution step, those two rate coefficients. And if, you know, the line of unity is a classic USP level A, where as the drug dissolves, it gets absorbed because it's dissolution rate limited, i.e. alpha is a very large value. And the other extreme is a reverse L-shaped profile where you get relatively quick dissolution, and then because it dissolves quickly probably, and then you get subsequent absorption. So you get this reverse L-shaped profile by virtue of, say, a small alpha value. So just by plotting these, I think you can gain some biopharmaceutic insight in terms of rate limiting steps. This, of course, assumes you're doing dissolution to mimic in vivo dissolution, which I think is always at least a goal in the back of our minds. So then here's just some data, and each of these is independent. I mean, a four-way crossover study was done at the time, but each of these formulations sort of independent of each other. Low pressure, fast, medium, and slow. And you can quickly see that this is a BCS class 1 drug. So the fast, medium, and low pressure are clearly sort of reverse L-shaped profile, low biopharmaceutic risk by inspection. The slow formulation actually is not that slow, but it is being pushed towards that line of unity a little bit. And you can fit the alpha value in this sort of thing, but it still wasn't very it was still permeation rate limited. Here's another example, ranitidine, a class 3 drug. But even the slow formulation is really not that slow. Expert formulators actually had a hard time making a slow formulation of this highly soluble drug. And then here's piroxicam. Piroxicam is a class 2 drug. It actually wasn't very, so it was, the slow formulation actually was dissolution rate limited. But what's kind of interesting is the formulators were able to change a class 2 drug from being dissolution rate limited to permeation rate limited. And that's sort of manifested in this plot here with the reverse L-shaped profiles for the more rapidly dissolving fast and medium and Feldine. And then sort of trying to anticipate that in the laboratory. Here's something we've been working on for a couple of years in terms of a dissolution permeation system involving a hollow fiber system that's hooked up to a USP apparatus. Drugs with known pharmacokinetics we've subjected to in vivo analysis as well as in vitro analysis, and we see good concordance for at least these simple formulations between what's observed in vivo clinically and then what this apparatus is able to anticipate in terms of biopharmaceutic risk. Okay, so those are some discussions about a high risk type of product and a lower type risk products. Thank you very much. Yeah. Are you back up, Hailing?
So I'm going to discuss the regulatory perspective for the in vitro dissolution technologies for biopredictive performance. Maybe we can skip the standard disclaimer in the interest of time. So what I want to do here is I already kind of spoil a little bit, right? We had a 2-day public workshop here last Thursday and Friday, discussing an updated biopharmaceutical risk assessment framework. So could we go to the next slide? So what I try to do here is really to give you a high-level summary, what have been discussed in that workshop, because I think it is very relevant to this session. Because so before I get into the detail of the risk assessment framework, I want to give a bit kind of summary what happened here a few days ago. So we had in-person workshop, 2-day workshop. We have over 200 attendees from industry, academia, FDA, and other regulatory agencies. In the 2-day workshop, we had 5 presentation sessions. Session 1 highlight the updated biopharmaceutical risk assessment framework. Session 2 focused on high-risk products. We had lots of interesting discussion around IVIVC and IVIVR studies for high-risk products. Session 3 focused on the medium-risk drug products. And we talk about some of the innovative biopharmaceutical tool for medium-risk products, including biorelevant dissolution in couple with PPPM. And then we next day session started with a low risk and a very low drug products, talking about what is needed and what is not needed. And then we closed the workshop with a future of dissolution testing. And then we also have 3 breakout sessions covered 6 very interesting topics here. Oops. Let me go back a little bit. So the topic A, we focus on the biopharmaceutical risk assessment framework, talk about the practical strategies for drug development and also regulatory decision making. The topic B focus on the high-risk product, talking about the risk characterization and the mitigation strategies. And then topic C focus on how to define medium-risk products, how to differentiate them from low and high-risk products. And then topic D focus on the biopredictive tool available for medium-risk products for risk mitigation as well as life cycle management. And the topic D discuss the no risk and very low risk product. We talk about a framework really based on the physiological and clinical relevance. And then finally, we discussed the strategy for future implementing the risk framework. So, oops, what did I do? Here we are. All right. So as I mentioned, I want to provide a high level of the what we have covered for this 2-day workshop in next 10 minutes. I know it's almost an impossible mission, but I will try my best. So traditionally, we emphasize or focus on developing dissolution method, asking discriminating ability against critical bioavailability attributes. So this approach has its merits. However, it has limitations. The biggest limitation is that those methods usually lack the relevance to the clinical performance. Also, most of those so-called critical bioavailability attributes are much better and well controlled by direct testing. So, and also identification and also control those so-called CBAs, you know, it tends to be very difficult, really identify them quantitatively and qualitatively. So the new framework, we're asking a different question. The question we're trying to ask is the dissolution truly, can the dissolution truly predict the in vivo performance? Can it differentiate products which are bioequivalent from those which are not? So the framework systemically evaluate 3 interplayed elements: the API properties, the physiological conditions, and then the formulation complexities. To classify drug products, all the oral drug products into 5 risk categories, from very low risk to very high risk. Each risk level dictates what evidence is needed to develop a biorelevant dissolution method. Let me fix this a little bit. Okay, here we are. All right. So, and I saw quite a few familiar faces in the audience. I know I'm basically borrowing some slides from my colleagues from last week presentation. So here is the 5 risk categories from very low to very high. For very low risk and no risk, our current thinking is that for those type of products, the determined risk for the drug product is the API physical chemical properties. Either is the API solubility or permeability. And then moving to the medium risk, in addition to the API properties, GI physiology condition can meaningfully impact the in vivo dissolution and absorption. And then moving from medium risk to high risk, in addition to the API physical chemical property, the GI physiology, the formulation technology itself really controls the bioavailability. And then for very high risk products, we take other factors into consideration, including some of the clinical factors such as narrow therapeutic index, early concept drugs, black box drugs, and also some of the really complex formulation technologies with layers of complexity. So as I mentioned, we use this kind of risk framework to determine what evidence is needed to develop a predictive dissolution method. So for a very low risk and no risk products, we are thinking the only development data going to be in vitro. In vivo data are really needed to support the dissolution method development. And then for medium risk product, we want to emphasize the role of biorelevant dissolution. We also thinking in vitro and some in vivo evidence along with the modeling and simulation approaches could be an effective way to manage medium risk drug products. And then for high risk products, we're looking more for IVIVC, IVIVR, in couple with some of the modeling simulation approaches. The in vivo evidence may have coming from the dedicated in vivo PK studies. So this is the overall of this framework. And then let's start with the low risk product. For low risk products, BCS is the foundational framework for classifying substances by their solubility and the permeability, and could be a powerful initial screen tool for biopharmaceutical risk. However, we want to emphasize that BCS criteria alone should not dictate the appropriate release testing strategy for the finished drug products. So for example, for BCS2A products, if they have a high solubility in small intestine where the absorption occur, we consider those are low risk drug product. Also a product with borderline high solubility or low solubility API may still consider as a low biopharmaceutical risk profile. And then we emphasize to develop fit for purpose release testing. The testing should be aligned with the totality of the evidence for the finished products. And then in terms of the risk assessment, we want to emphasize the API-driven performance and also emphasize the integration of the clinical data. If the API physical chemical property along with clinical PK data collectively support that the in vivo dissolution is not the rate limiting step, then the formulation really just serve as kind of, hold the tablets together. So for the cases like that, we're thinking maybe disintegration could be a replacement for dissolution test. And then once again, we want to emphasize release test decision should be based on the clinical PK data, physical chemical properties, and the in vivo performance. We'll talk about this kind of in vivo evidence. We're not asking the sponsors or applicants to conduct the dedicated PK studies. Really, this evidence could be from literature, from the understanding of your product. And then, and also even for low-risk products where we think a dissolution test is necessary, we going to move towards a standard dissolution method to support the release. And also we do think a standard dissolution method may be adequate to ensure the PK performance of the low-risk drug products. And then if you look at the ICH-M9 and ICH-M13P, this is really anchored based on the low risk and the very low risk drug products. For ICH-M9 BCS-based waiver is really based on the API solubility and the permeability. And then for ICH-M13B, for the biowaivers for IR products, we extend the biowaiver principle to the additional kind of either generic development or additional strengths, or even post-approval changes. With that, I want to spend a bit more time discussing the medium risk drug product. So the scientific basis for medium based drug products is those products have high or moderate permeability, but their in vivo dissolution is meaningfully impacted by the GI condition. For example, for a weak base BCS class 2B compound, the solubility change from stomach and to the intestine may be the key risk driver. And for a lipophilic drug, the biosauce mediated solubilization may be the main in vivo dissolution mechanisms. And then we also consider a clinically meaningful food effect is probably one of the clearest signal that the drug product is a medium risk. When AUC increase 40% or more and fat condition versus fasted condition, that clearly tells us that the GI conditions, including pH change, biosauce concentration, empty time, meaningfully impact the in vivo dissolution and absorption. And this phenomena cannot be captured by a conventional QC dissolution method, which is usually run under single pH, a buffer medium, and a non-physiologic condition. Once again, the boundaries between risk categories is scientifically defined. The key question separating medium from low risk is that whether GI physiology, not only the API physical chemical properties, meaningfully impact in vivo dissolution. And then the key question separating from medium risk to high risk is whether the formulation technology itself controls the bioavailability. So for medium risk of drug products, the formulation is usually is immediate release, conventional formulation, nothing fancy, no dissolution, no solubilization technology used. So when come to manage medium risk, when develop a predictive in vitro dissolution method, we envision that maybe start with the medium selection, could be a simple biorelevant dissolution, could be a simple buffer dissolution, and then choose the appropriate apparatus. If you want to use as a QC method, still USP apparatus were preferred. And then when you have a dissolution method, the key question here is your dissolution method is truly biodiscriminating, which means it can really tell or differentiate products which are bioequivalent from those which are not. Here we kind of thinking the validation can be done through either PBBM modeling or empirical IVR studies. Without this critical validation step, your biorelevant dissolution method is just another in vitro test. It will not become a predictive tool unless you anchor it to clinical outcomes. But for medium risk drug products, we'll consider the verification maybe can come from existing clinical studies, such as food effect study or comparative bioavailability studies. And if the in vitro dissolution method you developed is biodiscriminating, also is robust enough, you could use as a QC method. But we also recognize some of the more advanced biorelevant dissolution method may be not robust enough to be used as a QC method. In a scenario like this, we recommend a two-method control strategy. So we could have a approved QC method for batch release and routine testing, and a dissolution method approved for other purposes, such as for development, for life cycle management, for post-approved changes. Because the biorelevant dissolution presumably can capture the mechanistic kind of risk driving for the in vivo dissolution and absorption. But the key question here is to really bridge these two methods by demonstrating that your batch pass the QC method also fail within the safe space of your biorelevant dissolution method. So here I try to envision how this could be really applied, this medium risk framework to support a generic bioequivalence study for medium risk products. So if a product is identified as a medium risk, maybe we can start develop a biorelevant dissolution method, for example, FE-CIF, pH switch method, and then validate the predictive nature of those biorelevant dissolution methods either through PBBM or empirical IVR studies, and then use this validated biodissolution method to find a safe space to set a clinically relevant dissolution specifications. And hopefully this safe space can be applied to generic development, either for strengthening the biowaivers through the ICH M9 or M13B principles, or to support post-approval changes, and also for life cycle management. I'm on time, right? Okay. All right. So I'm going to very briefly touch upon the high-risk drug products. Because in the current framework, we are put all the extended or modified release, extended release product. We understand some of the delayed release products could be, treated as a simple IR products. But for the extended release formulation and the immediate release formulation with complex stabilization technology, we characterize them all in the high-risk products. In the interest of time, I'm going to go a little bit about the evidence hierarchy. So in the current framework, we recommend IVC studies for some of the extended release products, including BCS class 1 and the BCS class 3 ER products, prefer level A IVIVC study, point-to-point correlation. When level A IVIVC not feasible, we recommend multi-level C correlation or PBBM-based approaches. And so we also encourage to incorporate PBBM to provide mechanistic understanding and also use it to predict performance outside testing range by incorporating biorelevant dissolution data. So really the key principle is we try to use the IVIVC, IVIVR or modeling approach to find a bioequivalence safe space and how to use this safe space to develop generic high-risk drug products. I think we would like to have a further discussion with our industry colleagues. So with that, I'd like to provide a very brief summary. So dissolution is evolving from a quality control to a predictive patient-centric measure of bioperformance. The biopharmaceutical risk assessment framework provides a structured science-based approach to link dissolution to clinical performance. And we do see the opportunity, especially for the medium-risk drug products, to work with our industry colleagues to how to apply this framework for generic development. So with that, I'd like to thank my colleagues, especially Dr. Balgong and Dr. Xiaoming Xu from OPQ and all my colleagues in the DPQA 6 in OPQA 1 and the DPQA 12 in OPQA 2. I actually steal quite a few slides from their presentation. And then finally, thank you for your attention. Now let's welcome our next speaker. I believe it's Sivar. Yes, he's going to share the industry perspective, how to develop a truly predictive dissolution method. The Zhang.
Good morning, good afternoon, and good evening to the audience present here and also to the audience present online. Today, I'm going to talk about the perspective about what exactly is a true biopredictive dissolution media and what are different approaches the industry follows to develop such a biopredictive dissolution media. It's a standard disclaimer. Whatever I'm going to talk in this presentation, they are perfectly my views and they do not represent the statements or opinions of my organization. This is the brief agenda for today's discussion. I'm going to talk about various terminologies that are used in the dissolution, and I'm going to stress upon the quality control versus biopredictive dissolution, followed by certain approaches that the industry follow to develop a biopredictive dissolution, followed by few case examples where we have successfully developed a biopredictive dissolution, and then I'll summarize the entire discussion. So the day of the biopharmaceutical scientist as well as the pharmaceutical development scientist does not start and does not end without talking the topic about dissolution. What is the dissolution of my prototype formulation against the reference product? Is the dissolution slower or higher? And if it is different from the reference product, what is going to be the impact on bioequivalence? What kind of dissolution profile that my lower strengths have against the bioequivalent strengths? So all these questions will keep lingering in the brains of the biopharmaceutics and pharmacokinetic scientist with an ultimate objective to achieve the bioequivalence. Because dissolution is such a powerful tool, not only from the manufacturing quality as well as from the clinical quality. It is going to provide critical information into the in vivo performance of your generic product against the reference product. But when it comes to the dissolution, there are a lot of terminologies that are in the market, and we keep using these terminologies interchangeably. So some of these terminologies have already been established as a part of MCSC workshop that was conducted a couple of years ago, and I just would like to highlight the same. So when we talk about the quality control, typically the quality control dissolution is used for the formulation testing, and it lacks ability to reject the non-bioequivalent batches typically. And there is another set of terminology which is called as biorelevant dissolution. So typically the biorelevant dissolution is the dissolution condition that is mimicking the gastrointestinal tract in terms of pH, bile salts, electrolytes. And the biorelevant dissolution is not biopredictive unless and until you demonstrate it. Then there is another terminology which people typically use, which is called as bioindicative dissolution. Bioindicative dissolution is just going to provide a direction into the in vivo performance qualitatively, but not quantitatively. And the last set of resolution, which is called as biopredictive resolution, which is very powerful because it is not only going to provide the direction into the in vivo performance, but you'll be able to quantitatively measure this particular direction with the help of modeling and simulation tools. So often industry challenges is to have a quality control method which is biopredictive. Because as I mentioned previously, the quality control dissolution typically focuses on the manufacturing quality, whether my dissolution criteria is meeting for the commercial batches or batches at the production so that I can release the batches into the market. But often we don't evaluate the biopredictive ability of the dissolution because QC method is focusing on ensuring the completeness of the drug release in order to ensure that complete drug is available in the dosage form and which is getting released ultimately, but it doesn't provide a direction into the biopredictive ability. But when the QC method is also biopredictive, this represents a best case scenario. And that's why this kind of scenario we can classify into low risk. When it comes to the medium risk, the QC method is not biopredictive in both fasting and fed conditions. Then typically you need a couple of methods, one for the biopredictive assessment, other method is for the quality control, and that poses a medium risk. And when it comes to the aspect that you have a quality control dissolution available, but you're not able to develop a biopredictive dissolution at all. So that basically represents a high risk scenario. So it is possible that you may have a QC method which is biopredictive that represents the best case, but in case you are not able to do so, then you are landing up in a high risk scenario for your drug product. So what exactly is a true biopredictive dissolution? As I mentioned previously, it is going to provide a quantitative evidence into the in vivo prediction, and it need not be biorelevant, but the vice versa is true. A biorelevant method can also be biopredictive, and it should be able to discriminate against the multiple batches with varying clinical performance, and ultimately it can be combined with the modeling and simulation tools to provide a quantitative information into the bioequivalence or the in vivo drug product performance. The graph on the right-hand side talks about the trend in publications on the biopredictive dissolution over the couple of years. And especially when you focus on 2015 onwards, there is a sudden surge in terms of the publication. And thanks to all the initiatives by the regulatory agencies like FDA and EMA for coming up on PPBM and PPPK guidances, and thereby all the industry starts to emphasize more on the biopredictive dissolutions. So how do you develop a biopredictive dissolution method? Is there a standard textbook definition available for step-by-step approach to develop a biopredictive dissolution? Or where I should start, where I should end, and where I can conclude that I have a biopredictive dissolution? So this particular diagram talks about the development of a biopredictive dissolution from industry perspective. So whenever you have a product that is being introduced into the pipeline, first of all, you can consider the aspects like BCS classification, the formulation, whether it is an immediate release or a modified release formulation, pH dependent solubility or pH independent solubility, the in vivo Tmax, and of course, the literature available information. And based on these properties, you can have variety of methods available. For example, you can have a single buffer in pH 1.2, 4.5, 6.8 as a starting point. Sometimes you can also evaluate the carryover dissolution or a two-step dissolution, pH 1.2 or 4.5 followed by 6.8. You can also evaluate the biorelevant dissolutions, passive as well as specific media with surfactants, and sometimes you can also have alternative tools like residual analysis, which I'm going to cover in a particular case study. So once you have evaluated the performance of the drug product in multiple dissolution conditions based on the factors of API and formulation, then you're going to conduct a pilot study initially. And based on the pilot study results, you're going to get an insight which media is providing a direction into my in vivo performance. And if a further optimization of the media is required, you do that and you confirm the biopredictive ability of this media through subsequent pilot studies or using the pivotal studies. And this is the typical process flow that we follow in the industry for the development of biopredictive dissolutions. So with the next set of couple of case studies, I'm going to talk about how did we successfully develop a biopredictive dissolution for a couple of products. The first product is omeprazole DR ODT formulation. And the objective in this particular case is to develop a biopredictive dissolution that is mimicking both fasting and fed conditions based on the pilot study. And then ultimately we applied this media for the pivotal study to get a successful pivotal bioequivalence. It is a BCS class 2 molecule. Tmax is around 2 hours and 4 hours in fasting and fed conditions. The variability is more than 30%. and the Cmax and AUC gets reduced in presence of food, probably because of the degradation. And what kind of approaches we followed for biopredictive dissolution in this case is that, considering the delayed release nature of this formulation, we started evaluating the followed by medias, and we also evaluated the plain medias initially, but we did not find biopredictiveness. We also evaluated fasting and fed intestinal fluids, and surfactant addition was also not necessary in this case because there is a good amount of solubility for this particular drug in the basic condition. So this is the data in the followed by medias, 0.1 normal HCl followed by pH 6.8, representing the fasting case. And in the fed state, pH 5 followed by pH 6.8. We had a couple of data points from the pilot formulations in both fasting and fed conditions. And the medias were validated against the reference and test products in the fasting and fed conditions. And the prediction errors were also pretty less, as indicated by the graphs below. So when we combine these medias into the physiologically based biopharmaceutics model, we can clearly see that with the help of virtual simulations, the behavior of fasting as well as fed pilot batches are really indicated by the model. And especially when you focus on the fed condition, the lower T/R ratios for Cmax as well as AUC were also nicely predicted by the model. So in conclusion, for this particular case study, the carryover medias were found to be biopredictive in both the pilot fasting and fed condition. And we are able to get a medias validated against a failed bioequivalent study as well. The next set of case study I would like to provide is about alfuzosin prolonged release tablets. And the objective of this case study is development of biopredictive dissolution in fasting and fed conditions, and with an intention to get a reduced Cmax. So for this particular product, there are some adverse events associated with the Cmax. So we want to purposefully reduce the Cmax, but yet would like to attain a bioequivalence for the Cmax to reduce the event of adverse events. So this is a prolonged release tablet formulation, BCS class 3, and the Tmax is around 8 to 10 hours in the fasting and fed condition. So the approach we have taken for the biopredictive dissolution in this case is that it is a highly soluble API, and we evaluated the plain medias. And considering the pH independent nature of the polymer for this extended release formulation, we have seen that across the dissolution conditions, the dissolution behavior is similar. And we intentionally designed a test formulation with a slower release with the purpose of blunting the Cmax to reduce the risk of adverse events. So this is the graphical representation of the dissolution data in 0.01 normal HCl and pH 4.5. Around the Tmax region, intentionally for the generic product, we have reduced the dissolution slightly with an objective of getting the slightly reduced Cmax, but attaining the bioequivalence. And with the data coming from this biopredictive dissolution, when we model this behavior, we are sure that we are going to achieve the bioequivalence. And then the actual pivotal results also have indicated the same. So this is how we have used biopredictive dissolution in this case to attain a drug product with a slightly reduced Cmax, but yet bioequivalent drug product to get away with the adverse events. The next product is drug A IR capsule. So in this case, the objective is again to develop a biopredictive dissolution in fasting condition for a BCS class 2 containing IR product. And in this case, the class 2 molecule is manufactured with a standard technology. The molecule exhibits significant absorption around 85%, and Tmax is around 1 hour in fasting condition. The variability is 20 to 25%, and it is stable throughout the GI tract conditions. So in this case, we started with the plain medias, and we did not achieve a bio prediction in this case with the plain medias. So we switched over to the biorelevant media like passive media in this case with an optimized pH as well as the RPM, and it has given a good direction into the in vivo behavior. So this is the behavior of pilot 1, pilot 2, and the pivotal formulations. And especially in the pilot 1 and pilot 2 formulations, we see that the test product is faster in one case and the test product is slower in one case. And in the pivotal formulation, both the profiles are closely matching. So when we input the data of passive dissolution into the modeling and simulation tools, we are able to quantitatively measure the bioprediction, in this case, in the fasting condition. So the biopredictive media in this case is not the plain buffer, but the passive condition, and we are able to predict the performance of pilot and pivotal batches accurately. Now, the last case study is about drug B formulated as DR capsules. And this case study is a little bit special because the dissolution, none of the dissolution conditions were found to be biopredictive in this case. So it happens very often in the development that your dissolutions, whatever optimization you try with respect to RPM or pH, they may not be biopredictive. And in this case, in certain cases, you will have to find some alternative tools which can give an indication of the in vivo performance. That can be a kinetic solubility, or in this case, something like a residual analysis. So the molecule in this case is a BCS class 2, class 3 borderline, and the Tmax is 1 to 2 hours in fasting and 4 to 5 hours in PET condition. The variability is more than 30%, and it degrades in the acidic condition. So we evaluated a lot of medias in this case, followed by medias, plain medias, medias with surfactant and without surfactant, and also the biorelevant medias. And none of the medias have really predicted the in vivo behavior, especially in the PET condition. So for the DR formulations that undergoes degradation in the acidic conditions, the in vivo behavior is governed by the degradation. And that's where we came up with a novel tool called as residual analysis in pH 5 with 0.5% surfactant level. So if there is a degradation in this particular condition, that is directly going to reflect in the in vivo behavior for Cmax as well as AUC condition. So we have a couple of formulations tested in the pilot study, and we are able to get a pretty good correlation between the residual assay and the fed bioequivalence outcome. And we developed a simple regression based on this, and based on the regression equation that is developed, we optimize the polymer concentration, which is giving us almost equal amount of residue as that of the reference product. And with that, we are able to get a bioequivalence in this case. So in this case, the resolution did not work for the bioprediction, but alternative tools such as residual analysis has clearly provided the in vivo direction. And this also gives an opportunity to think beyond the dissolution for the biopredictive capability. So these four case studies that I have presented basically highlights the aspect of development of biopredictive dissolution. That can be plain media, that can be a carryover media, media with surfactant, or passive or passive, and in some cases, the residual analysis. So in summary, the trend of biopredictive dissolutions, we can clearly see that it has been increasing over a couple of years. And when you combine the data coming from biopredictive dissolution into the modeling tools, they provide a lot of information about the in vivo performance. And you'll have to evaluate the development of biopredictive media case by case basis, considering the product behavior, considering the formulation, considering the variability, and so on and so forth. So when you combine the biopredictive media with the quality control media, it really becomes a powerful tool, because you're not only going to ensure the clinical quality, but also the manufacturing quality as well. So I would like to thank Dr. Anuj, Dr. Rajkumar, and biopharmaceutics team in global clinical management at Dr. Reddy's. And I would like to thank my organization for providing the infrastructure and support. And this work presented here is collaboration from a lot of cross-functional teams who have contributed to the data and regulatory submissions. Last but not the least, I would like to thank CRCG and USFDA for providing me this opportunity to present my work. Thank you. I would like to introduce and welcome the next speaker, Deanna Mudie, Senior Principal Scientist, PBPK Research and Development, Simulations Plus. Thank you.
Thank you, Siva. Thanks everyone for your attention. I'm going to first talk about some general dissolution modeling considerations. I'll then go through 2 case studies and finally end with some take-home messages. So the first general consideration that Siva also talked about is that both biorelevant and QC dissolution methods may be suitable for model parameterization. So biorelevant methods may be preferred due to their physiological nature. However, quality control methods, which might use unrealistic volumes, might use synthetic surfactant, and also might be constrained to USP 1 or 2, are also potential for use in PBBMs. And that's when we combine them with use of mechanistic equations. So we can use these mechanistic equations to quantify these in vitro impacts, such as the impact of synthetic surfactant on drug solubility. And then we can use these equations to apply them to the physiological situation. So they really allow that in vitro to in vivo translation. The next general consideration is the importance of choosing the right dose to volume relative to your solubility in order to be able to describe the full dissolution profile, and also to be able to describe the influence of the entire extent of the particle size distribution or apparent solubility on performance. One good way to see if your dissolution data is suitable is to look at how many data points fall within that 20 to 80% absorbed range. So this profile has several data points here, although I would add a sampling time point pretty early on in order to get a full characterization of the profile. This example here is reasonable, but we're really lacking data before 80% dissolved, and that would really call into question the early part of the profile. The next profile on the lower right is non-ideal because, as you can see, most of the drug has absorbed before, or dissolved, excuse me, before the first time point. And one possible way to remedy this might be to increase the dose to volume in order to slow down the dissolution. The last curve has the opposite effect, where in this case, the dose to volume was much too high. And you can see here, only about 10% of the drug can be dissolved here. And now it's going to be virtually impossible to distinguish between different formulations or between different particle size distributions of the drug. So that would be a method that would not be easy to use to indicate whether your method's discriminating or to really characterize that profile. The next thing I'd like to touch upon are some of the equations we can use to parameterize dissolution. So this slide shows several options available in GastroPlus. These range from less mechanistic to more mechanistic. And when I say more mechanistic, what I really mean is the examples on the bottom have a much better or more inherent way to actually quantify the impacts of the physiological variables on the dissolution. And by doing that, we're able to much more easily translate in vitro to in vivo dissolution. However, all of these different equations can be suitable. It just really depends on the case. It depends on factors such as model purpose, rate determining steps to absorption, as well as the shape of the profile. Next, I'd like to move on to the first case study of omaveloxolone. And the key points here will be incorporating multiple release mechanisms, as well as the necessity of including mechanistic first pass extraction in some cases. Omaveloxolone is a highly lipophilic BCS2 drug. The most important things to know for this case study are that this was dosed in the amorphous form and that the amorphous solubility is relatively independent of pH. However, this drug is highly solubilized in bile salt micelles, so that's very important. The other thing I'd like to point out is that this drug is metabolized by CYP3A4 in the gut and liver. And then finally, it has a unique food effect. So the 150 milligram dose has a 350% increase in Cmax with a high-fat meal. However, the AUC only increases by about 15%. And this is unusual because normally we would see a similar percent increase in both the AUC and Cmax for drugs like this. In terms of parameterizing the release for this example, it's not just about the dissolution. So this drug product is solid amorphous drug particles in a solid blend in HPMC capsules. You can see in blue here that the solid blend has rapid dissolution, but when it's placed in the HPMC capsule, there's of course a delay in dissolution or in release due to that capsule opening time. So we wanted to independently take care of these two processes. We parameterize the dissolution using the Johnson equation, and the important inputs here are the particle size distribution of these API particles together with the total solubility in the presence of the bile salt micelles. The capsule opening was then handled separately using a Weibull function with the controlled release undissolved setup. And this allowed us again to use the model to determine the influence of capsule openings separately from things like particle size. The other thing that was important here was parameterizing the gut metabolism. So we had to parameterize the gut metabolism rate. So once these drug particles were dissolved and in solution and they reached the GI enterocyte, we needed to characterize the rate of metabolism. This was accomplished using the gut enzyme distribution factor, This is just the relative abundance of the CYP3A4 enzyme in different parts of the gut. Also use the Michaelis-Menten enzyme kinetic parameters specific to this drug, as well as the unbound drug concentrations in the enterocyte. What's important here is that you can see from this equation that metabolism rate depends on location within the gut, as well as drug concentration in the gut. And these are dependent on the dissolution rate and the absorption rate as well. And what we saw was that in the fed state, there was higher drug absorption in regions with higher CYP3A4 expression. So this shows percent drug absorption in the different gut compartments. You can see in orange in the fed state, we had much higher percent absorbed compared to the fasted state in blue. And when you look at this plot of relative enzyme expression in the different gut compartments, you can see indeed this area is the area with the highest gut enzyme expression. However, in the fasted state, we can see in the lower small intestine, the drug continued to be absorbed, whereas that wasn't so much the case in the fed state. And so what this ended up meaning was that there was a higher percent metabolism in the fed state compared to the fasted state. The reason for this higher upper intestinal absorption and gut metabolism was driven by the dissolution. So when we look at drug amount versus time undergoing different processes in the fasted state versus the fed state, you can see there was faster and higher dissolution in the fed state, and that was due to the higher concentration of bile salt micelles and the affinity of the drug for these micelles. In addition to that, however, I mentioned there was also more metabolism. And the net effect that this had was that there were similar amounts of drug reaching systemic circulation in the fasted and the fed state, resulting in the similar AUCs. And what's important here is that the 350% increase in Cmax that was due to this rapid dissolution and absorption, but again, with only the 15% increase in AUC, could not have been predicted using dissolution alone, and it couldn't have been predicted using the solubility alone either. It really took this mechanistic gut metabolism included in the model to get a full elucidation of the food effects. Okay, the next case study is on ziprasidone HCl. And the key points will be parameterizing dissolution using QC dissolution with surfactant, as well as incorporating salt effects. Here we're talking about a hydrochloride salt of a BCS2 weak base. And what I really want to focus on here is the solubility of the drug as a function of pH. So I mentioned we're talking about a salt form. So we can see the salt solubility profile with the dotted line, and then we can see the free base profile in the solid line. What's important is that to the left of the pH max at lower pH values, the salt is thermodynamically stable. However, to the right of the pH max, the salt has the potential to supersaturate, but it also has the potential to precipitate or convert to the free base form. And understanding these curves and what is actually happening is extremely important to parameterizing the dissolution test and understanding what's going on in vivo. Before I talk about the dissolution model parameterization, I wanted to spend a minute on the product particle size distribution equation, which is what we used. So this equation is similar to the Johnson model. It does have a difference of separately accounting for dissolution of drug that's free versus dissolution of drug that's bound to micelles. But what we do here is we solve for the radius, which we can assume as a polydispersed size distribution as a function of values that are known, such as solubility, dose, volume, and then micelle radius and micelle affinity. What makes this equation flexible is that the micelle affinity and radius depend on the micelle type you're using. So you can use it for synthetic surfactant, you can use it for passive-fessive or the in vivo situation, and that allows for the translation between those different scenarios. So we fit the PPSD equation using the NDA-QC dissolution method, which was in the literature. What was unfortunate, but often the case, is that not all the information was available. So we had to make some assumptions. The first assumption we made was that the dissolution occurs according to the free base solubility, or in other words, there was full conversion of the salt to the free form at this high pH of 7.4. The next thing we had to do was predict the solubility and the micelle affinity in sodium dodecyl sulfate because that measurement was not available in the literature. So we used an equation by Alex Abdeef to make that calculation. And then we also calculated the radius of the micelles. And once we did that, we fit the PPSD, and you can see it here where a two-bin particle size distribution adequately described the data. The next step we did was we verified that PPSD using a second medium, and this is pH 7.4 with a lower amount of surfactant. Unfortunately, the fitted PPSD underpredicts dissolution in other media. So you can see in phosphate pH 7.4 without surfactant, FASF and FESIV, we're not doing a good job of predicting the dissolution. So the reason for that, presumably, is that the salt did not convert completely to the free base. And instead, we need to take into account the salt solubility. You can see the actual data shows dissolution to a higher value. So next, we did some hypothesis testing to see if different salt to free base disproportionation levels would be able to describe the data. You can see in pH 7.4 with no surfactant, assuming that the salt converted 85% to the free base, described the data well. In FASF, a 65% conversion to free base worked well. And then in FESIV, assuming that there was no conversion or it remained completely as the salt, described the data. Of course, this is just hypothesis testing at this point. So what we'd want to do is further characterize these phenomena, perhaps do some PXRD analysis to see what forms are in solution. Also actually do some in-house measurements of solubility of the drug in the surfactant and further strengthen these hypotheses. But what this demonstrates is that it can be really complicated to fit profiles when you have a salt, when you have an amorphous form, where there's also precipitation or conversion happening. And then ideally, we would refit the PPSD and the other media as well. With that, I'd like to go over the take-home messages. So the first is that, we can get this, there we go. Quantifying aqueous solubility, micelle affinity, and micelle size in each dissolution medium, and also designing dose to achieve a meaningful rate in extended dissolution is critical for successful dissolution model parameterization. Biorelevant and QC dissolution have potential to be used to parameterize PBBMs with in vitro and to in vivo translation enabled by mechanistic equations. Modeling dissolution of supersaturating drugs that precipitate can be challenging, and it requires knowledge of form conversion in different media. When dissolution conditions and/or physicochemical properties in the dissolution media are lacking, assumptions must be made during initial modeling, but this does drive recommendations for further characterization and is another important reason that we do this modeling in the first place, because it really allows us to understand and interrogate our in vitro data. And then finally, additional processes besides dissolution often need to be accounted for in PBBMs to accurately describe the rate and extent of appearance in the bloodstream. So I mentioned capsule opening, also gut metabolism, but of course there are things like degradation, prodrug conversion that can also be important. I'd like to thank several people, especially Xavier Pepin, Sandra Suarez-Sharp, and Vera Lukachova for providing data, providing their guidance, as well as several other people that were authors on the papers and case studies. So thank you very much. And I'd like to introduce Ahmed Zidan, a senior research pharmacologist who will be making the next presentation.
Good morning and good afternoon for those who are joining virtually with us. And thank you, Deanna, and our speaker, for wonderful talks about the risk assessment, biopharm risk assessment framework that's proposed in last week workshop. So my slides today will focus also in one of the categories or one of the risk groups in the biopharm risk framework, which is the medium oral drug product. And risk assignments and the evidentiary packages that's needed to support the development of biorelevant method really change from a case to case. And over my presentation, I'm going to cover a lamotrigine case study and how we can develop this like evidentiary packages that support bioprediction of the solution in vivo. So this is like a traditional disclaimer that everyone is sharing. And this outline of my presentation. So the biopharm risk assessment framework, it is an architectured risk-based framework that is designed mainly to align the regulatory evidential requirement with the in vivo bioperformance. So usually, like as Dr. Lemberger have highlighted in the opening session yesterday, that the PSS classification still has a gap in defining the risk, relying mainly on the latency of API characteristics in terms of solubility and permeability alone. However, still neglecting a lot of inherent factors of GI physiology and formulation. And this can be highlighted in the medium risk group here. So the three pillars that this framework is depending on is mainly formulation design and complexity, ranging from IR formulation to amorphous solid dispersion and extended release approaches, and API physicochemical characteristics, not only solubility and permeation, but also particle size, distribution, and exchange and ionization all over GIT. And the least, but like the last but not the least, is the GI physiology, where the transition along the GI environment and the changing in regard to food effect and concomitant drug administration, like acid-reducing agent, can really alter the exposure and the in vivo performance. So for this one, the framework is categorizing most of the drug product depending on this risk and what type of evidentiary packages that may be needed to support each case. So we have two classes of the low risk group, the very low risk, mainly for PSC class 1 drug, where non-discriminatory dissolution method may be used, or low risk drug where has higher sensitivity, little bit higher risk with minimal GI impact. And in this case, quality control standard with discriminatory dissolution method may be used. And then the most like controversial group, which is the medium risk group, where there is like some sort of balance between the dosage form performance and its exposure to the GI physiology and transition in the GI. In this case, development of a biopredictive method require a full framework of strategies and methods to be integrated together, as Siva highlighted and Deanna highlighted in her presentation as well. And then the high risk group, including those who have narrow therapeutic index drug, where any change in the formulation or in drug amount can lead to in vivo variability. And the high risk group, including amorphous solid dispersion or extended release dosage form, and the use of IVIVC and develop a biopredictive method is a key here. So let me focus more on the medium risk now, which is the main focus of this presentation. In medium risk, so it is not defined by a single variable, but it's also it requires a whole harmony of all variables that's affecting bioavailability, as we call it, critical bioavailability parameters. And the main characteristics of this medium risk group is the interaction dependent, where the systemic exposure depends mainly on the interplay between the release rate in the gut and the physiologic environment, as well as the dissolution contingent here, where solubilization as a function of a changing in the pH is the one that is shaping or determining the shape of the PK profile, as well as moderate sensitivity to pH compared to the high risk and very high risk group. So the primary risk determinant in this case, it will be pH-dependent solubilization, of course, and the permeability profile, which is a harmony between the drug release and the drug exposure to the absorption site. So there is unwarranted balance that's happening there. So and in development of generic, we need to find a dissolution methods that can mimic this balance between drug release and permeation so that we can use a biopredictive method to justify the bioperformance. And of course, our method should be like sensitive to any failure of the drug product due to supersaturation or precipitation or dose dumping in this case. So now the big question is, what is the evidentiary package for a medium risk group? So it has a group of tools, as we call it, a control strategy, ranging from an understanding of the critical material attributes and process parameter of our formulation. We need to have a full understanding of what is the source of variability that's affecting the release profile, and then develop a biorelevant dissolution using, for example, biorelevant dissolution media, or mimic the shear effects that's happening in the gut. And then integrate this dissolution data with the BBK model as using the dissolution data as one of the input in validating and in creating the model. And then like establish an uncertainty analysis or sensitivity analysis here, where we are determining what is the most critical factor in whole situation that's coming from physiology, it's coming from the food effect, coming from acid reducing agent that are affecting the exposure. And then having like clinical prediction where we can simulate the clinical like outcomes across different scenarios, and then to establish a safe space. And within this safe space of the solution, any change in the formulation or in the release are not expected to produce an effect on the in vivo exposure. So this whole package is really a serious effort that's needed to be done for dissolution. And in some cases, it required to have like two dissolution methods, one for QC that's supporting just batch-to-batch variability and daily activities, and another one which is biopredictive to support any changes in the life cycle of the drug product. So let's go over a case study of lamotrigine API and how we can develop this package for lamotrigine. So lamotrigine here, it is a paradigmatic tier, and it's showing like the unwarranted balance between the molecular properties and the formulation kinetic. As an API, lamotrigine is class 2B drug, so it is weak base, and it has a pKa of 5.7, and it has a moderate permeability through the gut. So looking at the solubilization curve as a function of the pH, we find that lamotrigine, because it is a weak base, it solubilizes very readily in the gastric component, but once it hit the higher pH in the duodenum and proximal intestine, then a precipitation may happen here. If you look at the curve here, so this is the solubility curve, and this is the ionized form curve, and the ionization dropped dramatically. So if the dose number for this dosage forms that we are using is really high, then precipitation will happen. And this precipitation may be solubilized by the bile, or it may crystallize to other polymorph that is not solubilized by the bile, which is it will be a catastrophic event in this case. So in order to quantify the risk, we can use a rubric, numbered rubric, where we can give it numbers to identify this risk. Or we, for the sake of the slides today or my talk, I'm just going to just use a qualitative scoreboard. So I think for solubility, we demonstrated it is highly sensitive to solubility and the change in pH. And for permeability, it depends on the solubility of the drug, where it is it's soluble in different parts of the GIT. Lamotrigine is absorbed by passive diffusion. So once the drug solubilizes, it will get absorbed right away. So once there is no supersaturation or crystallization, then permeability will not be an issue here. In the gastric component, solubility is almost 4 milligrams per ml, meaning that if the dose of the drug is low, then there is no concern about permeability. But if we are using a really high dose, like 300 milligram of the drug, because this is different strength product, then we have a risk of precipitation. That's why for permeation or permeability, we are giving it medium risk. And for the release mechanism, it depends on how we develop our dosage form. If it is immediate release, so there is no control from the dosage form on the release rate. So in this case, we'll make it like low risk. But if it is extended release, it will be high risk. That's why for release mechanism, we give it medium risk as well. And for GIT transition, it is sensitive to pH transit, being sensitive to food effect, acid reducing agent, and residence time, and gastric emptying rate. So it is very sensitive. Now the question, is lamotrigine can be classified as class 3 or class 4 drug? I believe it depends on the formulations that it is formulated in. So if we are using immediate release formulation, so in this case, immediate release formulation, in this case, it will be medium risk because the drug here is mainly affected by the GIT solubilization and the pH-dependent solubility at each segment of the GIT. And the risk in this case is just to consider the pH change. However, if you are having extended release formulation, then it can be a high risk product, right? Because now the formulation is adding another factor in risk assessment that is complicate the situation or the in vivo exposure. So how now we can develop the 6 steps of the controlled strategy for lamotrigine? I remember that we mentioned the first step was understanding the formulation and critical formulation and process attributes, right? And this is what we are going to do here. For the extended release formulation of lamotrigine, it is using a matrix tablet that is based on hydrophilic polymers like HPMC, and it is coated mainly with a non-enteric coat, non-soluble coat. And it has 2 orifices at both sides of the tablet, and it is large orifice. But the main reason of having these 2 orifices is to make the release area or the hydration front and swelling and the erosion become constant during the release profile. So at the beginning of the release, drug get released from the whole tablet area, which is large surface area. That's why we have a faster release in the beginning. But as the release continue and the erosion of the dosage form is happening, then the rate of release start to decline. So it is non-linear release, like obeying diffusion kinetics. However, having this orifice here, it allow for more zero order and linear release of the drug despite of the extended release mechanism. So the critical formulation variables in this case that's controlling the in vivo absorption is the polymer viscosity, which is the one controlling the swelling and the erosion nature of the core, and the aperture dimension. So this is not an osmotic bump tablet. It is just like a release orifice, large, but there is no any osmogen or any pressure that's created inside the dosage form, as well as the coating integrity, because any failure in the coating integrity here may lead to a dose doubling in this case. So we have prepared this formulation into like different release rate using a variable amount of HPMC of different viscosity. So we have the fast release, which is using low viscosity and low amount of HPMC. And then we have a medium release, which has higher viscosity, but lower amount of HPMC. And then we have the slow release, which having a large amount of HPMC of higher viscosity. And you can see the in vivo profiles of these slow, fast, and medium formulation. So there is some proportionality of the release depending of the in vivo profile, depending on the in vivo release of the drug and its absorption. So we can say that we may use this 3 formulation to develop a BDK model and to validate it as well. So let's talk about the second part, which is developing an in vitro release method. So our distribution method here followed like a very similar strategy to what Shiva have mentioned in his talk. But we have used 2 like distribution apparatus, USP2 and USP2 and USP3. And for USP2, we have done it under both non-biorelevant condition and biorelevant condition. For the non-biorelevant condition, we just use buffer solution, and with it changing in the media as the pH propagate from 1.2 up to 6.8. And then for the biorelevant one, we have used a series of biorelevant media, starting from FastGEF, then FastSEF, and then FastCoF as well. And as long as it's changing. So we have like a gradual increase in the pH during the dissolution. And for USP3, we have used the same two media, biorelevant or non-biorelevant, but we have used the different amount of shears that's applied there. And as all of us know, in USP3, there is a limited volume in each dissolution vessel. However, we can use 6 steps of transition in the dissolution. And for this, we have find that the dissolution in USP2 was depending on the strength of the drug under biorelevant condition. And this is mainly because the drug precipitation as the dose number is increasing. So the 300 milligram strength didn't have like complete release here due to precipitation. However, if we are moving to non-biorelevant media and then adding surfactant to the media, which is allowing same conditions, then we have almost 100% release and comparable release rate from all the strengths. And for USP3, we found that the release profile was sensitive to the media composition. If we are using no surfactant in any of the biorelevant media, then we are having like almost incomplete release profile. But when we adding like surfactant to only the like simulated colon media, then we are having faster release and almost complete profile. And if we're having surfactant to all the biorelevant media, then the release is much faster and much complete. So this is a sensitivity to the composition of the release media, even if it is biorelevant and even if we are adding surfactant to the media. And similarly, the release was sensitive to the shearing rate or the dip rate in USP3. So increasing the dip rate was increasing the rate of the drug release. So for this, we have created a battery of dissolution methods that's ranging from a complete dissolution at faster rate or like incomplete dissolution at lower rate. So the next step was testing the sensitivity of these methods to the 3 formulations that we developed in the last slide. And we found that in most cases, like these methods are sensitive to the release rate from slow, medium, up to high rate. However, in USP3, we found that most of the release profile showed that incomplete dissolution. And this is, of course, because of the limited volume in USP3 and the supersaturation and the precipitation is happening when we are not adding surfactant to the media. So we have moved after that all of these dissolution data to the BBK modeling that we developed for lamotrigine. And for the BBK modeling, we have used the gastroblast for this case, and we consider the input parameter for the model using all like physicochemical characteristics of the API and general population parameters there, and the permeability and drug dissolution in all types of media that we have used. And we have used all of these as a parameter to train the PK profile. And we verified the simulated like model that we developed using intravenous data from literature. And we found that it has an immediate effect, almost to 2 microgram per milliliter at 0 minute. And there is decline. However, there is some variability there that found in the like in vivo profile for the predicted versus the actual in vivo profile. And then we have used the immediate release data from literature using immediate release capsule. And we find that there is some proportionality between the dose strength and the exposure in terms of EUC and Cmax. So we have used this data like the elimination, verification of the elimination constant in the BBK model and the absorption profile from the immediate release data here to work on the extended release profile. And for this, we have used the three strengths, 100 and 200 and 300 milligram. And we found there is a proportionality here of the exposure in terms of EUC and Cmax and even Tmax as well. And there is some prolongation of exposure as it is consistent with the controlled release kinetics there. So the second part is to deconvolute the in vivo data that of the three sets of formulations that we have prepared. And we have tested different deconvolution functions, as mentioned in previous slides. So we tried mechanistic deconvolution, numerical deconvolution, Wagner-Nelson, as well as low-Regelmann dissolution and stochastic dissolution. We find that low-Regelmann is the best to describe the in vivo release here. And this because the multi-compartment nature of the heterogeneous distribution in the body. And then we have used our formulation to validate the BBCOP model and to define the safe space. So for this one, we have moved all the data from USP2 and USP3 under both biorelevant and non-biorelevant condition. And this is coupled with low Regelmann deconvolution. And we have used the two approaches for internal validation, where we have built the model with three formulation, and we were only predicting the medium from the three, or even predicting the three formulation. And we have used external validation here when we build the model using the low release and the fast release, and we predict the medium release as it is not considered in the model in this case. And we found that we can have a nice IV-IVR here, and we were able to predict the low, medium, and fast release formulation with an acceptable internal validation of prediction error of less than 5%. And for external validation, the prediction error is still acceptable. It is below 7% or 5.4%. So our next step here is how we can define the safe space. For the safe space, it needs a simulation strategy. So we have used virtual patches and simulation runs to define the stress testing of the validated model using a virtual edge of cliff patches, which is defined by the failure in EUC and Cmax at the boundary of acceptability, which is ranging from 80% to 125%. And we have used a non-conforming batch or virtual batch that is not bioequivalent to the medium formulations that we developed. And based on our simulation, we found that the medium batch was non-equivalent. This is simulated batches showing that our safe space here can be validated. And the safe space or dissolution characteristics during the safe space has been determined at three levels. An initial phase here, which is amount released in 2 hours. And this is to account for the toxicity mitigation or the safety mitigation for the drug, where it prevents any toxic Cmax spikes or CNS-related adverse event like dizziness, ataxia due to high gastric solubility there. So this limit also protects the patient from any dose dumping. And then a medium point for specification, which is 6 hours. So it should be less than 45% here. And this is minimization of the supersaturation risk here and the drug precipitation in the gut. And then at 18 hours, it should be not less than 80%. And this account for the full bioavailability from the dose administered there, and it's ensuring the efficacy reservation. Now, our next point, can we rely on this developed model to mitigate or downgrade the risk? So let me first identify what do we mean by risk mitigation and risk downgrading or reclassification. For the risk mitigation here, meaning that I have enough evidence and I have enough tools that showing me that this risk is controlled so that in my scoreboard, I can give it a lower score of the risk. For example, the initial risk was 9, then I'm putting it down to 4, but not necessarily moving the drug product from one class to the other class. And this is what we meant by the risk downgrading here or reclassification. for the risk reclassification or downgrading, we should be ensured and have methods, and we have evidence, enough evidence to show that the drug is not liable to this risk anymore. And there is no risk in GIT, for example. So applying this concept to the lamotrigine case, we found that, yes, we can mitigate the risk if we develop this biopredictive framework for lamotrigine extended release formulation. However, we cannot like downgrade the risk to like lower criteria or lower tier here, because the drug is still like liable to any change in the pH and is still liable to food effect and acid reducing agent. So from this is also like the evidence that we described. And the lesson learned from my talk here, the solution is biopredictive only when it fits purpose and mechanistically interpretable and linked to in vivo performance. However, not simply because it is biorelevant or compendial, right? And the medium risk group here, it requires a totality of evidence approach that integrate the solution with PK, deconvolution function, IVR development and modeling and bioprediction and having a safe space. When we have a validated BBK model and IVC, it can bound and mitigate the risk. And this is support, of course, any post-approval decision-making and reduce uncertainty. But as we have shown here, it does not necessarily downgrade the risk. And the development of patient-specific quality standard for the solution is really a bridge here where we can translate the solution and modeling output into more clinically relevant specification. So at the end of my presentation, I would like to acknowledge all the project team from various OBQ offices and some of our staff here. They were with us during initiation of this project, and some of them had left. But thank you all for your collaboration on the project, and thank you for your attention. So I believe
Also would like to introduce our two virtual panelists, Dr. David Turner and Emilija. If you could join us and turn your camera on so people get to see your face. With that, I'd like to introduce our Q&A panel moderator, Dr. Ahmed Zidan, and Dr. Anisa Gordova will be our SME to screen the questions for us online.
welcome back from the cafe. And let's welcome our panelists in addition to our speaker today. So we have Dr. Andrew Babiskin, and he is the Deputy Division Director in DQMM in Office of Research and Standards. And we have Dr. Sandra Suarez-Sharp, and she is the Global Head Regulatory Strategies of Center of Excellence in Simulation Plus. And we have also Dr. David Turner, and he is a Senior Scientific Advisor and Head of Mechanistic Oral Absorption Modeling in Certara Predictive Technologies. So I believe David is with us online here. And we can start like with any in-person questions.
Hi, thanks for the wonderful presentations. So I have a question about the IVIVC development and the safe space concept, right? So say if you develop a IVIVC based on formulation changes, like James showed IVIVC based on changing amounts of a formulation excipient, but then you may have other issues. So for example, Ahmed showed multiple in vitro release studies changing not just formulation, but also some of the manufacturing variables, as well as maybe the release medium composition. So my understanding, and I'm not sure if this is correct. So my understanding is that If you have an IV-IVC based on one thing, such as formulation differences, well, that IV-IVC is kind of limited to formulation changes. So the IV-IVC wouldn't necessarily be sufficient to understanding changes in bioavailability if it's something like now you want to assess food effect. So ideally, you would have multiple IV-IVCs based on all those kind of aspects of concern. And so the key is to identify every possible issue that you could have that you want to use the IV-IVC in the model for. So basically, my question is that correct? Do you have to really have such a thorough model? Is it correct that the IV-IVC is kind of limited to the variables that you change to develop it?
Yeah.
Thanks, Coleman. I mean, I guess my basic understanding is you raise a very important point. Like what's relevant, right? Like in the case I presented, admittingly it was academic. I don't think anyone in manufacturing would confuse HPMCASL with M. Like I don't think they would probably make that distinction difference. I will say that obviously sources of polymer grades, I mean, it's not like there's a line in the sand between L and M, right? It's a gradation. And I think there's probably some internal controls and maybe companies even have further specifications for their own purpose, right? Companies have gotten burnt, right, by the grade changes unknown to them. So you raise a very important point. I certainly don't have the answers in terms of here's our technology. What should, there's 10 parameters that you can contemplate. But I do have the understanding that, an important concept is, so one argument would be, well, we control our manufacturing so well that we don't have any variables. But I don't know if that really holds water, in my opinion. I think it's possible where you have things under control. If there's not enough understanding that actually you can kind of go off track a little bit. But I don't have a special answer to your question. I think we all struggle with this. What are the relevant parameters? What should I be most worried about?
Maybe I could provide some regular perspective. Dissolution sitting at the intersection between the quality, the drug product quality and the drug product performance, right? Ideally, your dissolution able to discriminating against some of the quality attributes, right? That's the ideal situation. But the current framework actually shifts that emphasis. We emphasize more the dissolution should be relevant to the clinical performance. In other words, in most scenarios should be the PK behavior, right? And talking about the possible variables that could impact dissolution, when we have a product, it's really the confounding factors, right? It's not a single kind of attributes you're really gonna it's always come together to impact the product performance. So that part, I think, should be taken care of by the quality by design. You understand your design space. And then, the dissolution really should just reflect different dissolution rate when you come up with different variables with your formulation when you design your IV-IVC. And if you have that dissolution, that particular in vitro dissolution being linked to the in vivo performance, for example, by IV-IVC. So you have that kind of dissolution safe space with that particular dissolution method, right? So that dissolution method should be able to applicable to maybe some of the changes you're making down the road, as long as your dissolution data can be trained like within that safe space. And then we can very confident to see that formulation, that product is still bioequivalent to the pre-changed product. So that's where the concept's coming from. Would that can provide some kind of insights to you, the questions?
Yes, thank you. Yeah, thank you, both of you guys. Thank you.
Yeah, we don't have a lot. So I'll make this point, even though I don't think this was the direction you're going with your question, but you're almost talking about the universality, applicability of biopredictive methods. And if something was truly biopredictive, then you can throw anything in your magical media and you would know whether or not they would get the same response. But I just see that's not the case. There's some limitations to the changes that you make. And I think this is also a challenge for the alternative BE approaches that we're talking about yesterday, when you're trying to compare tests and reference generic and brand name product, which completely different formulation, could be completely different mechanism of release. Do we have that magic media where I can show bioequivalence? And that's why we emphasize so much on also then doing the modeling simulation approaches to understand both products, how you get there. But I think everyone's ultimately biopredictive. That would be great if we can just throw whatever we have in there, we know exactly what comes out, but there's going to be always some level of additional understanding that's needed in the incorporation into mechanistic understanding. So.
Yeah, I just want to add to all this wonderful discussion, right? I totally agree with what our panelists had said. 2 years to answer your question specifically, right? Can we use safe space? Let's say develop for an immediate release formulation based on particle size distribution to inform different levels of crystalline content, right? Can we do that? I think the answer depends on how much work you have put together, how much data you have used to develop your model. And sometimes I think it's insufficient. One more time, if you develop your safe space for particle size only, to be able to apply this to crystalline content, you need to have you need to pressure test your dissolution method in a conducting dissolution studies in a multi-variable level so that you can understand whether the method is able to also pick up for crystallinity and whether those dissolution profiles coming from crystalline content, hardness, and everything still are within that safe space. If those dissolution profiles are outside that safe space, means that your model, your safe space cannot be applicable to inform the design space for other failure modes.
Yeah, I believe all of our answers just put into the questions here, but it can be all totalized in a control strategy that you are developing here, with the control over the critical material attributes and process controls or process parameter that is really critical for the solution. Once you have this type of control and then you are prioritizing your risk parameters there, and then you develop the biopredictive method that is sensitive to the most critical parameter, what we call it critical bioavailability parameter there. So in this case, we are looking at the solution as a profile that's indicative of in vivo performance. And any change in this profile will come from these risk factors that you prioritized already, and then with appropriate controls over this parameter or critical bioavailability parameter, then the IVC can be applied in different situations.
I just have one more comment. And the question sort of baked in was, maybe multiple IVCs. Designing experiments in vitro is relatively easy, in my opinion, because we can just do lots of experiments. Designing in vivo studies is very painful because you only have 3 arms or something like that. So you have an incredibly limited number of variables you can study. And, doing one IVC is probably pretty good, but it's not going to cover all the parameters that you would prefer, I'm sure.
Okay, we have one more question.
Yeah, first of all, thank you for all really nice presentations and really practical use cases that made me think also about this question, which is partly also, I think you partly already touched it or answered it, but still, I mean, these new risk categories kind of open the spectrum. And I think what it opens is it's great that it opens a strive for further and more deeper understanding of products that we are developing. that we are developing. But it does make me wonder if we are now having this spectrum of like, let's say medium risk, that it's something that's new. What does it practically mean for like if you have a product that you then qualify as medium risk, you have understanding. So what does it mean in terms of the implications regarding waiver of studies? And you can maybe add a hypothetical example or just the direction of thinking. It's not necessarily a straightforward answer that I'm calling, but is it like, are we going in a direction for medium risk product? We're going to allow waiving of some studies, bioequivalent studies, like in different GI conditions or something like that?
So I can see just Emilija just joined us online now, and she is one of the session leads. So do you want to address this question, Emilija, about?
Yeah, sure. Thank you, Ahmed. Yeah, thank you for the very good question. We need to assess biopharmaceutics risk and categorize to know how much complexity our development bears, and for regulators, how much risk to patients approval of that product brings. Whether we can waive the studies and not do bioequivalent study, I don't see that happening. However, it can give opportunity, again, case-by-case basis, on how much understanding we gain on the product, how much experimental data, in silico data we have. It can help us perhaps maybe waive one of the studies or justify some change. So in my opinion, it speaks about the risk, but for industry, it just speaks about how much evidence we need to give to in our submission to regulators to be able to make their informative decision and approve product to the market, and how much flexibility we can get for post-approval changes, which are part of the life cycle of each product. Thank you.
Thank you, Emilija. So Sivan, you want to chime in?
Yeah. So I think in principle, I agree with what Emilija has mentioned. The risk categorization, like low risk or medium risk or high risk, talks about extent of focus that we need to emphasize on the product in order to get a specific outcome from the regulatory perspective. Say, for example, you have a failure F2 between lower strength and higher strength, and if the product is falling into the medium risk. And if you're able to follow the principles of medium risk and demonstrate that this dissolution profile differences will not have impact on in vivo, then you will be able to justify the waiver. Say, for example, if you have a high risk product and where the expectation is that you need to have a IV-AVC or PBBM to de-risk any kind of a dissolution differences and so forth. So if you have a high risk product with such kind of dissolution differences, then you need to apply an IV-AVC or PBBM to further de-risk the impact of dissolution differences on the in vivo performance. So the risk categorization is helpful for us to understand from an industry what kind of extent of investigation that we need to on a particular product to achieve a specific regulatory outcome. That's what is my opinion.
Briefly, yeah.
Yes,
Yes. It's an excellent question, and I'm gonna answer it from a ex-reviewer perspective, right? Once I was a regulator. So let's say a company sends me a submission that has and I'm reviewing the solution testing, right, and the impact. And that the solution is for a BCS class 2 product, right? And I see that the method is not discriminating for nothing. In addition, the control strategy for that product is far, and the design space is far apart from what was tested in the pivotal clinical trials. So in my view, that product is a high risk because does not give me confidence that the specifications that are being put forward are gonna be controlling the quality of the product and that they're gonna ensure consistent performance to the clinical trial. So therefore, unless the sponsor gives me data either that demonstrates that method is discriminating, and even more important, right? If you are setting specifications that are very wide, outside the ranges that were tested in the pivotal clinical trials. I like to know if my method is under or over discriminating. That's the key. If you don't have in vitro in vivo link, you will not know whether your method is over or under discriminating. And from regulatory perspective, you don't want a method that is under discriminating, right? So, and from industry perspective, you don't want a method that is over discriminating. So if you give all that perspective, PBM and everything, then the confidence of the reviewer increases.
Exactly.
They're able to inform a specification settings, and then gonna say, I'm gonna approve this product because the way I see it, the risk that I see now, they give me all this information is gonna be lower. And with confidence, I'm gonna inform specification settings to ensure the quality control, everyday quality control.
Yeah, thank you.
I guess.
Can I have a short follow-up? Maybe you can, it's just how does this tiering then connects to the high risk label that is in the M13A guideline? Does it connect in any way? Or is it like something completely different?
So I guess from a regular perspective, for medium risk, the even we're asking PPVM verification, the study requirements going to be much lower than high risk, right? We're asking maybe existing PK studies already in the literature. For example, we mentioned food effect studies or comparative bioavailability studies could be used to verify your model. I know you're more focused on how we can use this tool for bio-waiver, right? I think that there is opportunity. This framework is kind of brand new to FDA too. I think we want to continue the dialogue with the industry and with all offices within FDA to really how to best use this advanced tool, right? Using the M9 and M13B bio-waiver principles. If those more advanced dissolution methods truly can be verified to be biopredictive or biodiscriminating. I think there is definitely opportunity there.
Yeah, thank you.
Thanks.
Very quick to tie to his question regarding how this risk versus M13A risk. I think it's too early. I think there's overlap, but I don't think it's exactly the same because it serves for different purpose. M13A mainly look up the bioincreasing risk with regard to food condition. But this is I feel like there's some differences, but there's some probably overlap. But worse, just like what Hailing said, probably need to look further.
Yeah,
to say that.
certainly with the high risk in both these, there's a lot of similarities, a lot of overlap. Yeah.
All right. So one of the focuses of this meeting is BE. So if you think about, and all the things you talk about, right? Oh, I want to have some in vivo data to test my model against. So we're talking about like one of the focuses of this workshop is oncology products. Very difficult to get in vivo data. You have to do patient studies. You can't do can't really, as we've heard, do a lot of pilot studies. And so for those cases, and again, some of this is, thinking about cases where I'm developing a formulation. I tested in, different pH media. My formulation is the same as my comparator. I showed biorelevant media. It's the same in fed and fasted conditions. What, for just a normal low solubility product, conventional formulation, what are the risk factors that are being missed in that framework? And I think this goes, you say like, well, you should use mechanistic modeling to look at that. But the main input to the modeling is the dissolution rate. If my measured dissolution rates in, pH media and biorelevant media are the same, the inputs into my model are going to be the same. So, I want you to think about that situation. Like, what are the risks? And, that's the framework. What are the things that maybe aren't included in our models that might be different? Because that's, I think, the use case for some of these to say, look, I'm coming in and saying I'm having a proposal to avoid recruiting patients for my study. I have a formulation I think are very similar to my comparator. I have dissolution data that's very similar. My input to my model is going to be the same, right? So like I'd like to hear the perspectives of the members of the panel on that type of question, right? What do you see are the biggest risks in that scenario? What's being missed? And if there are risks there, then what's the framework for including those risks in our PBPK models?
Yeah, I think I want to add a one point to Rob's comment, right? If you have a full understanding of mechanistic understanding of your risk driver for in vivo dissolution and absorption, and that risk kind of drivers could be captured by your biorelevant method. And then, then we need to look at what's the remain or residual risk. If we feel like that residual risk is low enough, I think we should able to entertain the idea to use those in vitro method to establish bioequivalence between the generic, the test product, and the reference product.
So let's hear Deanna. What do you think?
Yeah, thank you. I think some potential risks in that situation might be around how the in vitro test was designed and if it's going to capture the important distinctions between the formulations. Some things that come to mind would be buffer strength, media composition in terms of interrogating differences. Maybe your test is a highly sync 50 millimolar phosphate test. Maybe the two formulations perform the same there. But if you maybe used a lower phosphate concentration, different pH, then you would start to see them pull away from each other. And I think the other thing is when you are looking at combining those formulations into a model, again, it's just, were you able to also interrogate things like the entire surface area for dissolution? I touched upon that with things like the dose concentration. So I think just making sure that you're making good decisions about the in vitro method itself before you feed it into the model and then assume it's going to be predictive would be important.
Yeah, so we have David. Do you want to chime in, David, about it?
Yeah, well, I agree with what Deanna is saying about the risks, related to your choice of medium. And following on from Deanna's presentation, if we, in the circumstances where we can break down or understand mechanistically what is going on, then we can perhaps discriminate in vivo where we can't discriminate in vitro, just depending on the sensitivities of the particular formulations. The limitation, as I think everybody knows, is that mechanistic modeling of dissolution is mainly restricted to particular immediate release formulations. So for those types of formulations, the mechanistic models can provide great insight. And I would like to make one additional comment here is that we're talking about have talked about biopredictive dissolution. And this is in the situation where we're plugging the dissolution into the PBPK model directly. In circumstances where we can mechanistically model and understand the dissolution, the in vitro dissolution does not necessarily need to be directly biopredictive itself, because if we're able to account for the factors affecting the dissolution with a mechanistic model, then we can reconstruct them in vivo under physiological conditions, and perhaps often end up with a different dissolution profile to what we had in vitro, because we're accounting for the differences in pH or whatever. But this is still a developing area.
Yeah. So just for the sake of time, I believe we have time for one online question. So Anita.
Speakers today. What fundamentally defines a dissolution method as truly biopredictive, particularly when key in vivo processes like supersaturation and precipitation are not directly captured in vitro? Maybe Dr. Sivacharan can start.
Yeah, I think a true biopredictive dissolution is something that is going to provide a direction into the quantitative direction into the in vivo perspective. So sometimes it may happen that your dissolution method is not exactly capturing maybe supersaturation or precipitation, but in reality it is happening under the in vivo conditions. Maybe in those cases, you start with, if there is an example of a weakly basic molecule that undergoes precipitation, you do some kind of a two-stage evaluation and then get an estimate of extent of preparation, how fastly the reference and test products are getting precipitated. But if a dissolution method is not able to discriminate, but then your subsequent pilot study is telling you that there is some kind of a precipitation followed by resolubilization based on the PK profile, you come back to the dissolution test and do some adaptations and then again resimulate so that you're able to have that kind of quantitative information. So putting it simply, you should be able to account for the in vivo processes that are happening through optimization of the resolution conditions. If you're not able to get a direction at the initial phase, after an initial set of pilot studies, you come back and re-optimize your resolution model and then confirm that whatever model media you have developed, that is truly biopredictive.
Well, any other panelists want to chime in on this question?
So there are specific questions for Dr. Deanna Mudie. Did you try to adjust parameter estimate the surface pH environment parameter or precipitation parameter or any other testing in GastroPlus to test alternate hypothesis in your case studies that you have presented?
Yeah, thank you. In that particular case, the surface solubility used for the salt form would have been, was the solubility that was used. So it did take the surface pH into account. For the free form, the solubility was so low, it probably didn't affect surface pH, although if it did, it would be in the wrong direction. But I think what would be really important in this case is to better understand things like what are the relative particle sizes between the salt and free form? You know, is the precipitated free form covering the surface of the particle? You know, all these things matter in terms of the relative surface areas of the two forms. You know, even is it happening at the surface or in the bulk? So I think there's a lot of really specific questions that need to be asked, but what I showed was an initial, you know, way to test the hypothesis and then really figure out what tests do we need to go to do to better figure this out.
Yeah, so thank you so much, Deanna, and let's thank all our speakers and panelists for the wonderful discussion. And still, the same subject will be discussed again in the small group working discussion at the end of the day, and will give us more in-person opportunity to interact and ask more questions. Thank you all. So we'll shift gear a little bit from the solution in our next session. We are going to start session 6 here, and it's focused mainly about the regulatory acceptance of alternative approaches. And in this session, we are going to explore the strategy for achieving regulatory acceptance when using alternative approaches to establish bioequivalence may be challenging in some scenarios. And this is talk behind those bioequivalence approaches like listed in BSGs, and what are the challenges that is like preventing adoption of new or emerging bioequivalence demonstration technologies there. We're going to start our speakers with Dr. Zhang. And Dr. Li is the Deputy Director of Office of Research and Standards.
Thank you, Ahmed. Good morning, good afternoon, good evening for all those who attend this workshop, either in person or online. This is session 6, which will be the last virtual session of the workshop. As you heard from Ahmed, it's going to focus on regulatory acceptance of alternative approaches. I think this is a topic that is in everybody's mind. And also it is very increasingly relevant when we try to address those challenging drug development questions that we are facing. So we have 3 core objectives for this session. First, we want to discuss the regulatory framework that governs the acceptance of those alternative B approaches, what the rules are, and where the flexibility lies. Second, we will ground that discussion in practical application through the real-life examples and also case studies that are going to be presented by the speakers. And third, we want to have an honest conversation about the challenges involved and also share all our best practices that both industry and FDA has learned along the way. By the end of this session, I hope you will leave with a clear sense of not just what the alternate B approaches are, but also how to use them effectively in your drug development program. And before we dive in, let's make sure we are aligned on what we mean by an alternate B approaches. So we know FDA do publish a lot of PSGs, product-specific guidances, on almost all the products in the Orange Books. And typically, standard B approaches are those going to be recommended in the PSGs that represent the well-established path according to our regulations, as well as the various general guidances. But also, more recently, those alternate B approaches, on the other hand, typically could be non-conventional and may fall outside of those standard recommendations we put in the PSG. And they may not be explicit describing PSGs, but that does not mean they are off the table. So you can still propose those alternate approaches based on your drug development programs, what problems you try to solve. The key question is always whether those approaches can adequately address the regulatory questions at your hand. And also, we also saw those, actually, I want to say, what does the regulatory framework actually already say in both our PSGs as well as general guidance? So you saw this disclaimer on all the guidances. We did say you can use an alternative approach if it satisfies the requirements of the applicable statutes and the regulations. So this phrase is the cornerstone. So you can always propose an alternative approach if you don't think what's currently in the PSG or guidance fit your purpose. FDA is not prescriptive on what that alternative approach is, but we do care about is whether the approach satisfies the underlying regulatory standard. And also both PSG as well as general guidance, as well as you heard from many speakers in this workshop, FDA encourage, this is not just a formality, but we encourage early dialogue, which is very important. As you heard through the workshop, and we will come back to that point. Probably you will hear that perspective again, early dialogue is very important. And here, what is the landscape of those alternate B approach? This slide just lists a few examples of the alternate B approach, which you may already heard from other sessions, as well as more examples you will hear from this session. There are several categories worth mentioning with regard to study design alternatives that include alternative study design, alternate comparators can be used, and also the bridging approach you have heard. With regard to modeling simulation, we talk about model-based BE, PBPK-supported biopharmaceutics decisions, and virtual BE, as well as very important about the evidence integration, including the BCS-based waiver, population COVID-informed, the model-informed evidence, and also very important is the totality of the evidence frameworks. These examples have been illustrated in the presentations in the past 2 days, early this morning, to address different challenges. And more will be presented in this session. So you will have the opportunity to see how they work in practice. So when you consider those alternative approach framework, there are 5 key questions probably you should ask yourself. First, what are the challenges with the current recommended approach? There should be a clear and legitimate reason why the standard approach is not feasible or appropriate for your product. Second, what problem does the alternative approach can help address or solve? The approach needs to be purposeful, not just because it's novel. Third, what is the scientific foundation and the regulatory framework to support the alternative approach that's going to fit within the existing regulatory expectations? Fourth, what data and justification will you use? The evidentiary package needs to be robust and coherent. And fifth, what FDA input do you need? This brings us back to the importance of early engagement, which I cannot emphasize enough. These 5 questions should guide your thinking from the very beginning of your drug development program. As you can see from the agenda, this session features an excellent lineup of 6 speakers from both industry as well as FDA, who will bring these concepts to life through some of the real-world examples and case studies. While these examples may not specific to oral oncology products, nitrosamine-related issues, or unavailable RDRS we talked about yesterday, however, the strategies and alternative approaches presented are going to be broadly transferable and applicable to a wide range of products as well as the therapeutic areas. Each presentation will address a unique challenge and also with a proposed solution. Together, they're going to paint a comprehensive picture of what it takes to achieve regulatory acceptance of alternative approaches. So let me summarize the key messages I want to take away from this session. FDA encourages the use of alternative BE approach when they are scientifically justified and appropriately applied. It is an active part how we support BE innovation in generic drug development. As you heard, this is the theme of this workshop. That said, success is not guaranteed simply by choosing an alternative method. It depends on aligning your scientific rationale with regulatory expectations from the outset. And that alignment is much easier to achieve when you communicate with FDA early. We have, as you can hear from this workshop, we have several mechanisms already available for that communication, from pre-ANDA meetings, the model integrated evidence, MIE meeting palette, controlled correspondences. So just use them for your benefit. Finally, I also want to note that FDA continues to invest in GDUFA science and research to address the challenges that make alternative approaches necessary in the first place. And we continue to update our PSGs to reflect our evolving science. And I also want to bring you to your attention that our annual GDUFA Science and Research Workshop will be held next month on June 8th to 9th, and where additional scientific and research priority topics will be discussed. So I encourage to monitor FDA's website to join that workshop, either virtually or in person. I will close with a few key takeaways I want you to hold on to when you listen to presentation in this session and beyond. The value of any alternative approach is not determined by how sophisticated or innovative the methodology is. It is determined by how well that approach addresses your specific regulatory challenge at hand, particularly across 3 dimensions: context of use, meaning whether the approach is fit for purpose; evidence integration, meaning whether the totality of your data tells a coherent and convincing story; and finally, the uncertainty management. We all need to be transparent on how you characterize and adequately address the limitation of those alternative approach. Keep those 3 dimensions in mind as you listen to the presentation today and when you engage in more in-person discussions throughout the small group discussion this afternoon. With that, I will turn it over to Dr. Jack Cook to start the first presentation. Thank you. He's gonna I believe, present online. Thank you.
So I'd like to talk about the use of covariates into establishing bioequivalence, and thank you for your consideration. Now, a couple of notes for the presentation. Most of the time I've used AUC to illustrate concepts. Everything generally holds true for Cmax. It's just that the math might be slightly more complicated when using a covariate. And I've simplified the example somewhat just to focus on concepts. Now, we run a standard two-way crossover trial. We use AUC as one of our key parameters, and under the assumption that if the clearance is same between the two occasions, period 1 and period 2 of the crossover trial, the ratio of dose normalized AUC will estimate the relative bioavailability. To assure the same clearance, we try to keep a lot of things the same, those being covariates. We try to use the same population for both, the same food, time. For other factors, we hope that randomization accounts for them equally for both formulations so that there's no bias. And when we run a crossover study, the variability in the estimate is can be called intra-subject or interoccasional variability. Unfortunately, there are times when you can't use a crossover design. Well, when would that occur? For drugs with very long half-lives, it makes crossover studies really difficult to retain people in the study. Safety concerns may necessitate studies in patients. You have to remember that healthy volunteers are actually a vulnerable population as they don't receive any medical benefit for being in a bioequivalence study. Patients may require continuous treatment, and that may be a limitation in the ability to do a crossover design. So what do you do in those cases? You use parallel group studies. They may be able to use, they may be able to introduce a bias due to the differences in those groups of the participants. You know, the little figure there is to show you that I differ from Jim immensely. Variability increases. You just no longer have intra-subject or interoccasional variability. You add on inter-subject variability, which takes the variability into the range of what would be considered high variability for a crossover study. With most drugs, the total variability is over 30%. So what are the solutions? Well, one solution is matching, and we try to do that in a lot of studies that have to be parallel group design. You try to match participants for covariates to reduce bias and variability. However, this matching often becomes a significant recruitment obstacle, especially if you have to do it in patients. Another alternative approach is to include those covariates, those things that you know might affect the pharmacokinetics of your drug, into the statistical analysis. I will say this is a work in progress, that it's not right now a standard practice, but I believe it offers a viable alternative. And as an example, I'm going to offer a drug interaction study of chloroquine, where chloroquine's being given with erythromycin or azithromycin, and we ran it as two parallel group studies. And in the graphs below, the two parallel groups are either one is represented by red circles and the other by blue circles. The goal here was actually to show that there wasn't a drug interaction using bioequivalence criteria. This was a product being developed, and the goal was to show that the improved efficacy with this regime was not simply due to azithromycin, increasing plasma concentrations of chloroquine. So we ran the studies one might typically do. Randomization didn't work. And what I mean by that is the heavier subjects tended to appear in one group while the lighter subjects appeared in the other group. And as you can see on the left, weight influenced the results both within and across groups. You see a clear trend that the larger you are, the lower your AUC was. Now, if one adjusts for those weights, you don't see that dependency anymore. And so we corrected chloroquine AUC simply by multiplying it by patient weight. And the graph on the right shows that there is no longer a weight weight influence on the outcome. And thankfully, the weight-adjusted results demonstrated lack of an interaction, as shown in this graph here, where both Cmax and AUC fell within the 80 to 120 confidence interval. Okay, so now some ideas on how one might implement the use of covariates. As I say, this is a work in progress because it's not routinely done. Well, one should consider doing this always from a prospective, not a retrospective point. The example I presented, it was done retrospectively. We examined the data through the normal method and without using a covariate, and saw that those two groups differed in their results, and had to do some work to find out that indeed weight might be the culprit. If you declare what you're going to do prospectively, there's a lot of things that ensures transparency, prevents bias, and enhances your credibility. And the big thing is it prevents a worry that you're just seeking an analytical technique to use the data to fit a desired outcome, where that outcome might be to show bioequivalence. I will note that ICH M13A suggests one can use covariates. In the parallel design study, they recommend that you should try to balance these covariates across group, but they also recommend those factors are also recommended to be accounted for in the primary statistical analysis. However, no further mention is how to implement that, and that's what I'll go through in the rest of the presentation. First, one would also want to consider the methodology. There are actually multiple ways to incorporate covariates. One could use a population model and to describe concentration time course. The pro of that, it's actually a more mechanistic way to introduce covariates in the molecule. For example, you might put weight or some other covariate such as concomitant medications as an influence on clearance, because we know that's how it affects it. The con is that with these intensive sampling methodologies, if you have a drug that has a complex absorption pattern, it may be difficult to exactly capture Cmax with a population model describing the curve. It has been utilized before. There is a reference given in a study for cyclosporin. Alternatively, one can use it in ANCOVA, like was shown in the drug interaction paper that I referenced earlier. Here, one directly uses the non-compartment analysis parameters Cmax and AUC. The downside of here is that it limits how covariates influence the parameters. It's less mechanistic, if you will. So what covariates does one use? Well, we're going to declare those prospectively. How can we do it? Well, the good news is that most drugs have data available as to what covariates should be considered. There are population PK studies. You can look into subject basis of approval to find out those factors that likely influence the pharmacokinetics. So you can grab that pool of covariates. I will note there's a couple of interesting things with that. While a covariate might not be considered important for a change in posology, it still may be influential considering our tight bioequivalence criteria. For example, if there was a covariate that changed the fraction absorbed by 20%, that's typically not one that one would say is clinically important enough to change the posology, but a 20% difference may be something worth putting into your model where you're evaluating the bioavailability of two different products, because there we have very tight criteria. One should also consider the incidence of a covariate. There are some covariates that may be large influencers, but they're not often seen in the population that you're going to study. And so it may be prudent just to exclude these, what I'll call rare covariates. Because we're going to use a statistical model, one should qualify the model. We have ICH M15 that talks about qualifications. And here, I won't go into the busy table, I just have it there for your use, that simulations will likely be very useful to establish the model's bioequivalence. You could compare two formulations that, say, have a 10% difference in AUC and Cmax to ensure that you'll have adequate performance. You can also use that to get sample size. You can also use it to show that if I have something that's outside of what should be declared bioequivalent, that the model does indeed show that. I love such simulations because you know the truth of the matter as far as your simulations go. So you can help establish or qualify the model through simulation. Now, a consideration for getting regulatory acceptance, well, the obvious, and you just heard it, since this approach has little precedence, you should prospectively engage with whatever regulatory body in order to discuss what you want to do. Maybe less obvious is that if this methodology has enough application, and I charge the group out there for considering this, you might want to consider drafting a regulatory guidance for the FDA. Personal experience notes that they're very open to receiving guidance suggestions, and here you could help fill in the blanks with some of the suggestions I have. What's adequate qualification of a model, maybe a more rigorous criteria for which covariates should be included, and what the package should look like. So I wish to thank you for your attention. Happy to answer any questions during the panel discussion, and I will let the next speaker come to the podium.
Thank you, Dr. Jacques. So we can move to our next speaker. He is also attending virtually, Dr. Yasvanth Ashokraj, and he is the Director of Biopharm and Pharmacokinetic at CIBLA. And he is going to give a presentation about model-based bioequivalence approach to resolve bioequivalence failures from outliers.
First of all, I wish to thank FDA and CRCG for providing this opportunity to present in this forum and steer some discussion on the B failures which are originating from the outliers and to resolve it through the model-based bioequivalence. Before I start, I would like to give the standard disclaimer that the views and opinions expressed in this presentation are solely personal and do not necessarily reflect the organization where I am affiliated with. So I'll be walking through the regulatory remedies for the outliers and the model-based bioequivalence and its introduction with its application. And we'll move on to a case study on the MBB, where what we have done on the population pharmacokinetic model fitting, validation, and uncertainty estimation, followed by the simulations, conclusions, and the scope. From the regulatory standpoint, before the ICH M13A guideline, there were several different approaches being suggested by various regulatory authorities. But once the ICH M13A guideline have been accepted by various and all the regulatory authorities, now the requirement of handling the outlier is being fixed now, like when the extreme PK values should not be removed from the B should not be removed from the B study solely because they appear statistically extreme. Only the well-documented protocol violations are allowed with the pre-specification in the protocol. Further, there can be certain exceptions. I would say only one exceptions where very low or non-measurable exposures are observed in the study. That can be determined by having the AUC of that particular subject if it is less than 5% compared to the geo mean of the AUC of that product, then that subject can be removed from the study and then calculate the bioequivalence. And this can be done only for only one subject per study. And more importantly, as unlike earlier, the redosing studies are not accepted anymore. With this given constraint, the outliers which do not fit into these criteria would require a redosing of the study to really show the bioequivalence of the products. Coming to the model-based B framework, this framework is the collaborative work of the US FDA and the Uppsala University, where they have outlined how model-based bioequivalence need to be performed in a stepwise manner. First of all, the B data need to be modeled, and with the fitted model need to have the treatment effect on the absorption parameters such as Ka and bioavailability. And further, it need to be further estimated for the uncertainty estimation using the methodologies which are outlined in the paper like SIR, bootstrap, or model averaging. And once this is done, the sampling from the model model and the parameter uncertainty need to be done to generate multiple population simulations, that is B replicates, and then compute the individual Cmax and AUCs from those B replicates and calculate for the Cmax and AUCs. And the distribution of those ratio mean need to be computed, and then the 90% CA of the ratio of the mean will be ultimately calculated to conclude the B. And the B is concluded when the 5th and 95th percentile of the Cmax and AUCs were well within 80 to 125. This model-based bioequivalence applications, model-based bioequivalence have been implemented for several applications and has been published in various publications. For example, this can be implemented for sparse sampling B designs, and it can also be used for time reduction in long duration B studies, such as long-acting injectable. But this was under the discussion during this workshop, and it was demonstrated to have a better prediction for the steady state, and also can be used for highly variable drug and drug products for better designing of the study and also for sampling strategies. Coming to the case study, the product what we have taken for this particular case study is a low risk product as per the ICH M13A guideline. And the B design is a fasting crossover in healthy male subjects. The B outcome was a passing for AUC and failing for Cmax marginally. And there are three reasons to select this particular case study. One is the product is simple IR, and we know that there were no any formulation related impact which would impact the which would have been driven the outlier. And the second is we had extensive B experience in this product where we developed for multiple markets, and we did multiple B studies because of that. And we had a data where the same formulation was redosed, and we'll be discussing that part in the later part. So this is the data what we have got from the reference and test product. As we can see here, there is one particular subject which showed a very low pharmacokinetic profile, and we couldn't find any plausible reason for the same. And as we can see here, when we remove this particular subject's data from the data set, we could see that the study was passing for the lower CA where it was failing with the outlier. And also there is a significant decrease in the inter-subject CV from 23 to 12. So, and we know that this could be the right data because of the experience we had in the other studies. Now, coming to the model development, this B data was modeled to a two-compartment model, and we found that the intra-subject IIV was over the clearance, VP, and the KA with the additive error model, and there were no demographic effect on the AUC and Cmax. And the demographics what we had were the BMI, height, weight, and the normal covariates what we the demographic parameters what we had used to have in the B study. From the validation point of view, we could see that the observed and fitted data were very well aligned, and you can see that particular point from the T-bar ratio, 90% CI, and also the inter-subject variability. The model was found to be well validated, and we can see understand from the GOF of the validation parameters. Like we can see the model adequacy and the standard diagnostic tools with this standard diagnostics tools where no systemic bias was observed, and we could see that there is a good agreement between the observed and the predicted parameters through this VPC profiles, except for one particular time point of 36 hours where there is a slight bias was observed. And the model uncertainty estimation showed that the estimates are the PK parameter, the fixed effect parameters showed the very low RSE or acceptable RSE, and the lower and higher CA were in acceptable range where it is centered around the estimates. And this was done by bootstrap method, non-parametric bootstrap method using 1,000 samples. Coming to simulation scheme, as outlined by the B frame, the model-based biochemical framework, we did 100 B replicates, 1,000 B replicates, and the number of subjects were fixed to 27 as per the original B study, And the Cmax and AUCs were derived from simulated continuous PK profile. This is the recommendation. Otherwise, in the NCA analysis, we pick up only the Cmax points from the observed profiles. And dosing and sample durations as per the original B study, and period and sequent effect not included as covariate. Further, as per the requirement, the type 1 error rate and the powers were computed. Basically, the ratios were, the type 1 error were analyzed at the ratios of 0.8 and 1.25, whereas the power was assessed at 0.9 ratio respectively. Further, the B determination, as I mentioned earlier, the 5th and 95th percentiles were calculated from the density distribution of the geometric mean ratio and formed the 90% CI. And we concluded B when the 90% CI for Cmax and AUC were within 80 to 120. Coming to the simulation outcome, the type 1 error rate was achieved and it was found to be less than 5%, and the power was achieved at 84% level. And we can see here, compared to the original outcome of the B through the NCA, the MBB outcome showed that the products are bioequivalent, as we can see from the 90% CI lower, which is well within the B limits. And this was verified, this information was verified in the sense we could see that the redose study also showed that the products were bioequivalent, and that's how we confirmed from all aspects that this methodology could work well. There are a couple of challenges, I would say, in my opinion, that where in this methodology, the covariates are not sufficient. There will not be sufficient enough if the variability is more actually. And when the absorption profile is a complex profile where multiple peaks are there, then we could face for implementing this methodology for this purpose. To conclude, model-based bioequivalence methodology can resolve the outliers-driven B failures in a low-risk product, and this has a potential application in oncology products requiring patient-based B designs where recruitment challenges are significant. And also, we believe that it will be useful in B risk assessment of high-risk products in case of redosing is an option for the sponsor. I would like to acknowledge Dr. Vijay Yuturi from Pumas A and SOFAS for his scientific discussion, and my colleague Vaibhavraj Bamre for helping on the model development, validation, and simulation for this work. Thank you for your attention.
Thank you, Yasvanth. And we have taken a lot of your time before lunch. I know we are hungry. So we are going to go to our last talk before lunch, Dr. Zhang, and he is a master pharmacologist in Division of Bioequivalence 1, Office of, I'm sorry, Biostatistics. Yes, Office of Bioequivalence. And he's going to talk about regulatory overview of alternative approaches and lessons learned from submissions and pre-engagement forums.
Hello, everyone. I know you. This is lunchtime. We are a little bit behind schedule. I'm the only one standing between you and the lunch, so I'll try to be on time. So I will provide a regulatory review of alternative bioequivalence approaches based on our review experiences from submissions and the engagement forums. This is a standard disclaimer. I'll skip it. My presentation mainly focuses on 3 areas. One is regulatory experiences in alternative BE approaches. And secondly, I'll present a few alternative approaches and case studies. And lastly, it's about I'll present a few engagement forums. I'll conclude my presentation with a brief summary. Over the years, we see a lot of we see substantial increases of alternative BE approaches. So for some alternative BE approaches, we already gained a lot of review experience. They are already well accepted. For example, BCS-based bioweavers, reference-scaled average bioequivalence, and alternative comparators when no RDRS is available, but there is an alternative BE product such as capsule versus tablet. These approaches are well accepted now. And over the years, there are also emerging approaches and submissions. We already heard a lot about modeling and simulation approaches such as PBPK, population PK, adaptive study designs, IVC, especially to support bioequivalence, when this IVC is linked to PBPK. And also there's new bridging studies when no RDRS or alternative BE product is available, but there's a non-BE product exists which can serve as a bridge. And there are also other emerging approaches. So so now we are getting more and more applications for these emerging approaches. Our confidence increases when we review more and more these cases. So let's start with the TTRR-TT study design. This is a popular study design. Yesterday, you heard about two talks presented this study design. So it's especially useful for drugs with a long half-life and high variability, especially when a steady state BE study in patients is recommended. It has a lot of benefits. It has shorter study duration, reduced sample size, potential to enable the use of RSA-BE approach. Yeah, we have reviewed. So this study design has been proposed by industry through controlled correspondence, pre-ANDA meetings, bio-INDs, and ANDA submissions. Reviewers are getting more and more familiar with this study design. One specific point I want to mention is that a PSG include the RRTT, TTRR study design, but that study design is not the exact study design I mentioned here. So it has some differences. So just want to bring to your attention. So during our BE bioequivalence review, we closely collaborated with Office of Research and Standard and Office of Biostatistics. Here, I listed a few data analysis considerations for you to consider when you design your study and conduct data analysis. First one is statistical method. This is not a typical fully replicated study design, but when you conduct statistical analysis, you can still use the conventional use a fully replicated study data analysis method. The second one is how to evaluate the steady state. So here, since so in general, you don't need to evaluate steady state for the third and the fourth, for the second and the fourth period, if the first and the third period already reach steady state. And yesterday, there were questions about unequal carryover. Yes, that's one issue for this study design. So this should be evaluated during your data analysis. Another point is usually for this study design, it has a smaller within subject variability than regular replicate study designs. But this, in general, this is not a concern because it's not inflate the variability and also, so this is not a concern. Another point is about pooling. So usually this study is conducted in multiple clinical centers with very few subjects. When you so in FDA statistical guidance, we commonly recommend you pool smaller clinical site into a larger clinical site for the purpose of statistical analysis. So the pooling method should be pre-specified. The last one is about group effect. If the study is conducted in multiple groups, so we recommend you to include group terms in your statistical analysis. Second one is adaptive design. We see also increasing number of studies with this adaptive design, and it has a lot of benefits. It can increase statistical efficiency, reduce patient exposure, and also reduce risk of study failure. And the portable method C is the most commonly used adaptive BE design in under submissions. And based on our BE review experience, here are some recommendations when you plan to use adaptive design. So first of all, pre-specify the use of adaptive design in your protocol. And secondly, provide a scientific justification for selecting an adaptive design supported by either published literature or your own simulation results. If you use an adaptive design developed through simulation, please ensure it is applied only to the simulation scenario for which it was developed. For example, if your simulation is based on two-way crossover, so it may not it is not applicable to a replicate study design. Lastly, use a theoretical TR ratio that you use in your simulation rather than the observed sample TR ratio to calculate the stage 1 power. Now let's switch gear to another very popular alternative approach, PBPK modeling and simulation. So ICH M15 was recently published. The general principle applies to PBPK modeling and simulation for BE purpose. And here I will focus on the model influence part. Model influence is the intended weight of the model outcomes in decision-making, considering the contribution of other relevant information. So it means if you already have a lot of other evidence to support your decision, then the model plays a minor role. But if the pivotal information is lacking, the model will play a major role. So I will give two case studies. The first case study is related to low model influence. So this is an oral extended release template. The PBPK model was used to address non-comparable dissolution data between the non-Bow strengths and the Bow strengths. We already have the data, see the data with acceptable fasting and fed BE studies, acceptable formulation proportionality across strengths. The only issue is about this is related to non-comparable dissolution data. F2 is less than 50. But we already have, for this specific case, we have a good mechanistic understanding of the release mechanism. So they observed the slower drug release from the Bow strengths attributed to same condition. This can be mitigated by comparing multiple units of the non-Bow strengths to a single unit of the Bow strengths of test product. So even though applicant proposed to use, actually they submitted a PBPK model to address this non-comparable dissolution scenario, but so since we already have a lot of other pivotal evidence to support to address this non-comparable dissolution issue, so this model is considered has low influence on our BE determination. So recommendation is engage with the agency as early as possible to clarify expectations regarding model development, validation, and role in establishing bioequivalence. The second case is also a very unique case. It's a high model. It has high model influence. It is a ER tablets. For the whole for this whole workshop, you heard about a lot of PBPK model and simulation, but all of this model, PBPK model development and validation is based on Cmax and AUC. But for this specific, the concern is about the T-lag. So the applicant tried to use PBPK IVC model to justify a potential T-lag issue. So compared to the RLD for a Weaver strength. So this T-lag issue is maybe a clinical meaningful. We already, for this case, we have evidence to show adequate fasting and Fed-B studies, acceptable formulation proportionality, comparable dissolution between non-Bow strengths and Bow strengths. But the pivotal, but since we have this T-lag concern, the pivotal in vivo or in vitro evidence is still lacking. And also the applicant's PBPK IVC model was not developed to address this T-lag issue. It can detect differences for Cmax and AUC, but it cannot really detect the differences for T-lag. So that's a major issue with this PBPK IVC. It's also for the fit of purpose use of PBPK. So for this specific case, in addition to Cmax and AUC, applicants should also develop a PBPK method that can be sufficiently discriminatory to detect T-lag differences. Again, engage with the agency as early as possible to clarify expectations. So for this specific case, if applicants contact agency early, we may point out them to develop a PBPK model, especially for a T-lag issue. Okay, so yeah, here is some engagement forums you can use. For pre-ANDA submissions, there are product development meeting, pre-submission meeting, model integrated evidence meeting, control correspondence. And there are also a few post-ANDA submission engagement you can use, such as mid-cycle review meeting, enhanced mid-cycle review meeting, post-CRL scientific meeting, and control correspondence. In summary, scientifically sound alternative BE approaches are encouraged, and the agency's confidence in certain alternative approaches continues to increase with additional review experience. The role of a model depends on its influence on the BE determination. If you plan to develop a model to address a specific BE question, we highly encourage you to engage with the agency as early as possible. Lastly, but not least, I want to thank my colleagues, Utpal, Yi-Hsien, and Fang for their comments on my slides. And also I want to thank Wanjie for their statistical expertise. I borrowed some slides ideas from her previous presentation. Yeah. With that, I conclude my presentation and enjoy your lunch. Thanks.